Welcome everyone to the fourth day of our workshop on skill enhancement on multi-omics data integration and data analytics from 27 September till 30th September 2022. This workshop is being organized by Center for Computational Biology and Bioinformatics, MIT University, Uttar Pradesh, Noida campus, in collaboration with Ensemble Team, European Bioinformatics Institute, Cambridge, UK, Tauber Bioinformatics Research Center, University of Haifa, Israel, Pine Biotech, USA, and Lucida Corporation, New Delhi. So our first session for today starts with Poly, data-centric ML Ops platform by Ms. Kritika Gaonkar, Lucida, New Delhi. Ms. Kritika Gaonkar is currently working as a senior solutions architect, identifying bioinformatics infrastructure and data needs for biomedical companies and implementing solutions to drive customer success is data. Prior to joining Lucida, she had the opportunity to work as bioinformatics scientist at Children's Hospital of Philadelphia. She works on collaborative projects with investigators in Children's Hospital of Philadelphia and in institutes participating in pediatric brain cancer research. As a part of the center, she develops and applies workflows to assist investigators by performing genomic analysis and visualizing data from CBDTC and other publicly available datasets. I now request Ms. Kritika Gaonkar to start the session.
Great, thank you, Payal. That was a great introduction. Great, happy to have everyone here. Today we'll be talking about Elucidator, but please feel free to ask me any questions after the talk as well. In general, multi-omics data analysis or about Elucidator. I'll share my screen because we have a few slides to go through, but I'm just going to use them to talk about the general trend that we see in biotech and R&D and the need for data-centric platform that Poly can provide. So the agenda for today is to address the challenges that are inherent to biomedical data, either publicly available or proprietary data that a company will generate. And we strongly believe in Elucidator to have a data-centric approach to data, just so that the value of the data is retained and that you can reuse the data for more analysis than it was earlier intended to. We'll go through the data infrastructure on Poly and also have a few comments on the future outlook of data-centric platforms, which will helpfully streamline the quality and insight generation in R&D, preclinical and clinical areas. So as you might know, biomedical data grows every year. This is a representation of how it has grown since the 1900s to this year, and it's an exponential curve. And we see this trend a lot in public domain. So you have seen this probably in your lifetime as well, that earlier a lot of data would be maybe 10 to 20 data sets. But now if you're going around or more than 1,000 data sets, then you are publishing in great publications or like having the impact in that community. So you've already seen that a lot of data is useful to drive R&D and research. But we at Elucidator know that a lot of data is already publicly available and useful, but it's just a problem that the data, even if it's available, is not usable. You can't search it effectively. So there is a trend where companies and research teams will generate more data just so that it's an easier way to do that. Maybe they have the funding to do it, so they generate more data, but it's time consuming and also takes more effort to manage that much amount of data that they generate in-house. So in Elucidator, we want to address this key problem that either like publicly available data sets or proprietary need to be on a platform that makes data the most valuable asset. And so then we can provide that to all stakeholders in the company that want to use it and reuse it for different questions that they have. And you can imagine like for a data to be produced, there are multiple stakeholders. So there's a bench scientist who's actually generating that data. So maybe they're sequencing it. There's like a lab experiment that's generating the data. So they are the main or like the starting point of the data generation, and they have specific asks from the data. So that would be, is the quality good? Was the protocol followed? Is the data usable for this current need? They don't really think about how other people will use the data directly, because that's not their job title, basically. They are there to generate the data. They have the expertise in generating the data and following protocol and analyzing the data for that specific need. But a lot of other people are required to make that data usable and valuable. So for example, you might have a bioinformatics scientist who's actually preparing, transforming, aggregating all this data so that it can be used to do some analysis and derive insights from it by a data analyst or a bioinformatician. And in the middle, we also have cloud architects now, because a lot of people know the value of using cloud architecture for data, but it requires another specific expert in the company or your research team that can handle all the warehousing needs and storing the data efficiently without costing you a lot of money. So you see there's a lot of people involved in managing data today. And all of them have specific viewpoints for the data. And if it's not essential for the company to make the data available for all of these stakeholders, then there will be silos for that data. So for example, the data generated by a biologist, maybe the bioinformatician has access to it, but a data scientist in that group will have to go through all these hurdles to find out who the bioinformatician was, who the biologist was, where the data resides, is it clean, is it valuable, and then get to the actual insight generation. So if there is a way to like make the data available to all the stakeholders, then this journey from generating to insight will be efficient. And you can see that now the data would be available to all the stakeholders and not be siloed. And it's not just like the 4 people that are generating and storing and deriving insights from the data, but there are also other people involved in actually funding that experiment, like data custodians or leadership that are funding that experiment, that need access to it in terms of like if they want to send out a report to their board members or have a presentation to identify where next to put their funds on. So a lot of people in the company and research teams need to have access to the data, even if they're not like directly, using that data hands-on. Somewhere in their job profile, having access to that data is essential for the company and also for research to happen efficiently. So this is a quote from Vas Narasimha. He's the CEO at Novartis. And he has a key point to talk about here, which we believe a lot of our customers and a lot of research teams across the um, world also face, where you want to like identify or derive insights from all this data that is available. But the key point here is that a lot of time is actually spent on cleaning the data. So previously, when I worked in Children's Hospital of Philadelphia, we tried to do this, where I was asked to look for publicly available data sets for pediatric tumors. And I could find out some of it from like looking at literature, looking at geo, and came up with a list. But it took me like months to identify if the data can be processed, if the data is available. If it's available, is it clean enough so that we can aggregate that information together? And it was a never ending task. And I still don't think anyone has completed that. Now it's been 3 years since we had started that project, just because it was so messy that we didn't feel like we could invest more time into cleaning that data. And instead, we just created more data to help us with our research. So this importance of having really good data to make your ML studies actually useful is a key point in biomedical data research that everyone at some point will feel in their careers and in their companies. Um, I'd like to stop here to just get like some interaction, or like, have you, previously felt this pain point, or like, have you seen some of this in your own experience?
I think the participants can write it in the chat box because they do not have speaking rights. So
Okay, sounds good. Well, I'm sure you feel the pain at some point.
yeah, I can agree to that. So the cleaning the data is the most difficult task of any data that is provided, whether it is from publicly available resources or even coming from an hospital setup.
Right.
Pharmaceutical data, it is very difficult to even look at the data at first.
Right. Yeah. Just like to do any like generic plots for bar plots or pie charts, you have to do so much cleaning that it used to just take us days to get to that point. Um, Great. So on Elucidator's platform, which is called Poly, we have a solution where we have centered the platform to make the data the most valuable asset, like I spoke about before. And that is mainly because we have a data-centric approach to data, and we use ML to scale up that approach. So that's why Poly is a data-centric ML ops platform. Uh, and what that entails is basically adopting uh, the code first approach to all steps of the data's journey. So for example, ingestion, we make sure that there are ETL pipelines to ingest data so that the code is reusable. Everyone knows how to reuse that data. There is standardization in the pre-processing as well as customization in terms of metadata or harmonization of metadata. So all of those are coded, Dockerized ETL pipelines. So there's standardization of the data ingestion from public sources or proprietary sources. So that makes sure that the data that you see today and maybe 2 months from now look the same and have the same harmonized metadata. The other aspect of this is data warehousing. Like we spoke about, that people generate a lot of data, you can store it in one way, but there needs to be cloud architects and data scientists who actually support the warehousing in an effective way that helps users to use that data for ML studies. So it needs to be clean, it needs to be linked to the source, indexed efficiently so that you can search for those data sets using some key terms. And in general, improving the data access and reuse by the stakeholders that we spoke about before. And finally, for the insight generation, um, that also is code first. So it's not, our approach is to enable the users to use the data for consumption through code by default. So we have a lot of user experience and interviews that we do with our customers to understand how they want to consume the data. And we've come up with very flexible and usable Python package to query the data that you will see in a few hours from now from some of our colleagues. And that just enables users to slice and dice the data as they want flexibly without having to stick to the specific, maybe the definition of a data set that we have defined or any of the stakeholders have defined. It allows more easy consumption through code. So these three points help adopt a data-centric approach on Poly. And as an example, I'll walk you through this use case where, so imagine you're generating a lot of data in your lab. So you would store it in an Excel sheet or a CSV file. Maybe you have some specific ways that you're storing it, following a file structure. But maybe your teammate is storing it in CSV files and naming the file formats and folders separately. So now if a data scientist joins your team, he will have to understand from both of you, like how you've stored the data set, what it contains, how you've named it. And there's no standards that he can follow to like automatically use that data generated to set up a ML study on. So the ingestion for that ML scientist will often be manual. It'll require a lot of cleaning, and maybe he'll have to hire an intern to do that. So there's a lot of time that is wasted on getting to that clean step where they can actually do the insight generation. But on Poly, our platform enables that because, like I mentioned, we would set up ETL pipelines to standardize that file format. So for example, you and your teammate have generated data in CSV files, Excel sheets, etc. Our process would be to standardize that data and add harmonized metadata so that all of them follow a specific standard format. So here we're using GCT, which is a generic format for tabular data, but it would be a standard format where anyone else who's trying to access the data maybe a year from now still knows what the contents are, how they've named, and what those metadata mean, because there is like reproducible ETL pipelines to go back to and identify all those steps. And because of the standardization, we can also automate this process. So the data scientist can stay back and like look at his ML's results instead of actually spending all this time in ingesting and aggregating the data. And curation, as I mentioned, is driven by ML on our platform. And you might have to be really trained on good data to identify those labels. So we have a lot of manual curators who actually are domain experts and will annotate those data sets using double-blinded studies and use that as our training set to scale up the ML models. So here we are showing drug, dose, frequency, and strength that are labeled in these two free text. So for example, we have some drug here, the dose and the strength. So what our ML models will do is use these labels from our domain experts and try to scale up by using weak supervision and do labeling so that you can improve the curation for data sets that come along maybe a week from now. And you can scale it up because it's automated ML model. And you can also have them, the manual curators, recheck the data on the platform. So we allow the scaling up of curation, but we also allow manual curators to recheck the curation of the data sets, just because we know that manual models will also have some caveats. We want to enable users and our internal curators to actually improve the data in an iterative manner without having like a one-step process for curation, because curation is an ongoing process. You learn new things about the data. You learn new things about your ML study needs. So we have this option to have like automate and also scale up the curation if required. For warehousing, again, like I mentioned, there'll be different file names, folder names, and you have to ask multiple people of what they actually meant by this not helpful name folder, which kind of leads to data being siloed and like kept by certain people in certain directories that maybe everyone doesn't have access to, which is not very useful for a company who wants to be like data driven, or a research team that wants to use a lot of data that they've generated. So following a standard format and a schema on the platform allows this more insight generation or data-driven insights, because now everyone knows that the file is stored in a specific format. For example, here, this is the counts, the genes are the rows, and the samples are at column here. And every metadata associated with the sample is stored in the same file. As you can see here, there is some drug information, dose information. And for this whole data set, we have additional information at data set level. So we know that this data set comes from human. It has 1,288 samples, and it's a GCT file. So there are ways you can slice this data set, which is predictable because now all the data sets that you've seen before and that you'll also see a month from now will always be GCT files. So if you create your ML study, you'll be able to automatically use this file structure and add into your database of training or test data sets effectively. And then the final part is consumption, which is basically what you want to do with data. So if the data set is not standardized, it's not queryable, you have to talk to a lot of people to get to that data. It really hampers the quality, speed, and the actual insights that are derived from that data. You can imagine that if you are tired from asking around and cleaning the data, you can only produce that much result that are of good value. Because research is also a very iterative and ongoing process. So for example, if you had wasted less time cleaning the data, you would have put more information and more thought into the analysis, the models that you've built, some QCs. So we believe that consumption is only valuable when you have the data that is clean and accessible and reusable. So on our platform, with the standardization of the file formats, the querying capabilities that we have on the platform that you'll see in the workshops later today, you'll be able to get to a higher quality, improve the speed and insights of the data sets, just because now you have flipped the time that you would have wasted on cleaning the data to actually deriving insights, doing more meaningful discussions with your collaborators as to what that result mean, and integrating with other useful tools that will give you more insights into the data set. And all this would be code first approach, mainly because this is how we have set up our platform, that the code first approach is the default to allow efficient data querying, standardization, and also allowing users to develop those ML models effectively. And in general, this is what our data platform looks like. And this is just a recap of the previous slides, that maybe you have some not so helpful file formats and folders on the platform. Through our ML ops standardization and curation process, we will be able to harmonize the data, automate the ingestion, and store it in a way that you have the data accessible through programmatic or UI-based, or also integrate with your most lovable applications that you have been using previously, but it was more manual effort that time. But through this curation, it would probably be more efficient to do that. Sorry about that. Yeah, and finally, before we go to a platform demo, I wanted to tell you about the Data Fair conference that we are having next month. So please try to join us there. I'll put the link in the chat after the talk today. We have a lot of great speakers in that conference. So please try to join us there. We also have a lot of positions that we've opened in different teams. So you can also check out those positions if you're interested in those. We can definitely talk about it more. I have also shared my email here if you have any questions, which I again will share in the chat. And I'll do a brief demo of the platform. We will have our colleagues talk about it in detail in a few hours in the hands-on workshop. But in general, our platform is for data. So we have thought about like storing data, using data efficiently on the platform. So this is what we call an omics atlas, which is basically a space or a component where we aggregate a lot of data, which could follow a specific tissue of interest or disease of interest, or these are all the data sets that you have used in your research and find useful. So what we would do is, this is publicly available data sets, but you can see that we've imported a lot of data sets from different sources. So for example, import is a source where a lot of immune-related studies are stored, but these are all lab-generated data sets. But geo is a lot of omics data types, like transcriptomics or ATAC-seq, etc. And all of these can be supported on the platform together. So in this omics atlas, we have stored around 1,700 data sets. And we also support a lot of different data types. As I mentioned, we have a lot of lab measurement data, but we also have transcriptomics data, gene dependency scores from DepMap, proteomics data, drug response. You might have analyzed or known some of these data types before. So you'll be interested in like exploring this omics atlas. We'll share the login to this account for the workshops. You can take a look into all these data types. And like I mentioned, we also can store or curate the disease terms here. So that's how you can actually aggregate and visualize what the distribution is for each disease. So a lot of this is arthritis, but we have Parkinson's, some normal data sets, adenocarcinoma of the lung. So it's a lot of data and useful data. So if you want to look into details about this data sets, you would go here, you click on view details. And this is where you would find all the data sets. And for each data set, we do curation for some key domains or biological entities. One of them is disease, which we use mesh ontology to normalize. We have organism, tissue, drug, and data type also reported here. And this is mainly so that the data is now findable and queryable using a standardized term for tissue. So like Homo sapiens is called Homo sapiens across all these data sets, instead of calling it human in one, Homo sapiens in the other one, or H.sapiens in the third one. All of them are called Homo sapien. And you can do quick queries now on the UI and more complex queries that you see in the workshops using these terms. So for example, if today I want to search for transcriptomics data sets for Parkinson's disease, I'll be able to do that like within a few seconds here. And I already know 345 data sets. are of use to me because they are tagged with Parkinson's data sets and are transcriptomics. And for some of these data sets, you might want to actually see more details. So from here, you know that they have studied a lot of diseases, or the study describes a lot of these diseases. And from the description, you also know some of the details here that these are patient data. And if you want to look for more details, like clicking on view details, without reading the paper too much, you'll be able to get insights into the data set and the protocol used. So for example, like I mentioned, you know, these are Parkinson's disease patients. There are 25 samples, and there's a link to the literature if you want to read more. And because of the curation, you already know that 36% of these data sets are normal, and 64% of these data sets are Parkinson's disease. And there'll be multiple data sets will be where if the same comparison will occur, that you can do quickly via querying for data sets on the Poly platform. Because everywhere, wherever it was Parkinson's versus control, it will be annotated as Parkinson's and normal respectively. Instead of calling it control or non-patient or healthy patient, everyone will be called as normal in our disease field. It just allows for a more structured and easier accessibility of the data set. And you also know that the tissue is substantia nigra. So without even having to read the paper, you know a lot of this by looking at the view details here. And of course, you also have the actual matrix here so that you can use this data set to actually derive insights, which is also supported on the platform. So by clicking analyze here, you'll be able to see some of these consumption methods. So Discover is our Shiny app that we've developed. And the other one here is a notebook that we have for exploring transcriptomics data sets. And both these consumption methods or some of the consumption methods will be shown in the hands-on workshops. So I hope you join those calls. So yeah, I think this is a good overview of the platform. So I'll take a pause here and take some questions from the attendees.
Okay, Kritika, there is one query in the chat box. Actually, it's not a query, it's a comment that says sometimes it becomes tough to even retrieve the data. Currently, I'm facing same issue of data retrieval. Process of retrieval is really cumbersome. So do you have any tips for her to do it and how she should be working through it?
Right. So I would say the best way to look for data sets or like retrieve the data sets is to follow some of the literature that your disease or tissue of interest have developed. Because a lot of times people will do some of these aggregation studies. So there'll be like 1,000 samples for pediatric tumors, 1,000 samples for breast cancer that they've studied. You will directly get like a list of a lot of data sets that are useful and available for public use. And if you don't find those, if you're like working on like a very niche project, I would ask you to like know more about the publicly available databases. Some of them, like even I am still learning about, but a lot of these public databases are very useful, but because of like less awareness or just because of the way that these databases have been set up, there's not a lot of like awareness about them. So I would encourage you to like learn about those databases and know how to access those databases um, automatically or through the APIs that they have. There are also useful tools that help you search for these data sets online. But again, like you have to clean the data set. So like retrieving and accessing the data set is the first problem. But cleaning the data set, storing it, making it usable is a whole another problem, which we all have to go through. We have to face those problems and learn from them. But yeah, I think you can start with some literature searches that should help you.
Yeah, thank you for the great advice. I think there are no further questions in the chat box. It was great listening from you, and I hope the hands-on session will give more clarity to everybody how it should be. What is the path to take to when you learn all these data curation things?
Okay.
Yeah, yeah,
Yeah, I'll also share some of the links that I spoke about today about our conference and any positions that we have open.
sure.
I would be happy to hear from you guys. I'll also be available throughout the hands-on workshop. So if you have any questions, then you can let me know as well.
Sure. We'll get back to you with the questions if there are any more.
Okay.
Thank you. Thank you, Kritika. Over to you, Payal. I think Gaurang has also joined us.
Yes, ma'am. I can see that Sarah has joined. So shall we start with the next
Yes.
talk?
Yeah.
Yes,
Hi,
I
Gaurang.
joined.
Yeah.
Yes.
Are we good to go?
Yeah, we are good to go. Can I share my screen?
Just
The
give me 2
presentation?
minutes.
Sure.
I think you are, yes, you are a co-host now. You can share.
Okay, cool.
So we'll just
Thank
give
you.
a quick introduction of yours and then we'll start. So give us 2 minutes. Yes, over to you, Payal.
Thank you, ma'am. So our next session is on patient-specific selection of drug candidates to target pancreatic cancer by Dr. Gaurav Mahajan, UC Data, New Delhi. Dr. Gaurav Mahajan is a machine learning researcher at UC Data. He pursued his PhD in physics from the Indus University Center for Astronomy and Astrophysics, Pune, and his postdoc is from the Indian Institute of Science Education and Research, Pune, in computational neurosciences. His research area includes mathematics, mathematical modeling, scientific computation, and data analysis and visualization using Python. I now request Dr. Gaurav Mahajan to start the session.
Yeah, just let me share my screen. Thanks for the kind introduction and the opportunity to present at this workshop. So Kritika has given you folks an in-depth introduction to our MLOps platform, Poly, and how it's enabling our customers to get maximum value out of their data for life sciences R&D efforts. I'll be talking a bit about exploring ML applications of the data as part of a recent initiative at Elucidata, in which I work. But before I get into that, I would like to begin by providing some general context. So as you would be aware, mostly machine learning continues to be adopted across a wide spectrum of biomedical applications. Some of these are foundational in nature, like the recent breakthroughs with AlphaFold predictions of protein structure, but some of them are also on the clinical application side, like discovering new treatments or supporting decisions in the clinic. So this graph shows that collaborations at the interface of ML and pharma R&D have been steadily increasing, and they're set to grow further. And Elucidata is, of course, an example of such a collaboration. Machine learning in general provides a versatile set of tools to make sense of all kinds of complex biological data across varied problem areas, and that's why it's increasingly popular. But with this promise also come challenges to using ML effectively for solving biomedical problems, while avoiding the problem of sunken costs and human harm. So that's the broad theme that I will be covering in this presentation. So what are these biomedical applications of machine learning? So broadly, they fall into two types. Like you have supervised learning models, where the goal is to sort of make a prediction based on some input data that you feed the model. So this could be biomedical or biomolecular data from, say, gene expression measurements. And the other class of algorithms is, of course, unsupervised learning, where you basically feed large data sets into a model. And this is more of a discovery type approach, where the goal is to sort of discover underlying patterns in the data distribution, and then, for example, identifying subgroups within a disease cohort. And sometimes the outputs from such an unsupervised model could feed back to a supervised model and help train those models by, for example, by providing new biological features. These applications can be like something very basic, like predicting small molecules that are likely to bind to a target protein, but they can also be about predicting clinical phenotypes like survival of a patient diagnosed with a disease, or diagnostic testing, or predicting treatment outcomes. So one of the pain points to useful application of ML is aspects of the data itself. So one quirk about biomedical data generally is that the data set sizes are very small. So compared to what you hear about here in the context of, say, vision or language models, where you have access to millions of data points, and biomedical applications, typically you only have access to a few hundred or a few thousand data points. Secondly, the number of features is very large. So if you consider gene expression measurements from a multi-omics experiment, that would be like 20 to 30,000 features for the human genome, of course. So the number of features can be much larger than the number of data points. So most biomedical ML problems lie within the higher dimensional setting. Another issue is that data for biomedical research problems can be of uneven quality. So typically you would like to pool data from multiple sources to increase the size of your training sets. But pooling data presents its own challenges. There could be a possibility of bias or noise in the data. So essentially, like, one can find several examples of such data issues plaguing the performance and utility of ML applications in the biomedical field. So I just wanted to highlight this with two quick examples from the recent literature. So one of them is like a recent meta-analysis or review of thousands of papers which came up with models to predict COVID-19 from medical images using machine learning, deep learning models. So what these folks did was to basically review the literature on this topic and filter publications based on the quality of their models, their methodology, the metrics. And the sobering conclusion of the study was that none of more than 1,000 such models were fit to be deployed in a clinical setting. And one recurring issue with these models was not that the models were wrong or inadequate, but the data they used was of poor quality. There were biases in the data. Basically, in a hurry, people try to put together data from different sources without applying proper quality control. So the main drawback with most of these studies was that the data quality was too poor to draw reliable and useful conclusions. Yeah, so I started by highlighting a couple of examples which illustrate how.
Sorry to disrupt. The screen is a little tilted. I believe it's not just my end. Half of the screen is visible and the rest half is. Yeah, now it's fine. Now it's fine.
Oh, that was crazy. Okay.
Yeah.
Thanks for letting me know.
Yeah.
So I was highlighting some examples from the recent literature which suggests that data quality can be a big bottleneck to successful application of ML. So that other example I wanted to highlight was this recent paper which claimed that a deep learning model does a good job of predicting enzyme activity from protein sequences and the substrate molecule information. But later work by other groups showed that you don't need a deep learning model here. Like even a simpler model can predict enzyme activity with comparable accuracy. And another issue that people pointed out was the data set that was used for training the model. The information about enzymes from the Brenda database had a lot of misannotated enzymes. So about 20% of the enzymes on Brenda happen to be misannotated, and these could significantly impact the performance of these models. So examples like these illustrate that ML projects are not just a model problem, but they're also a data problem. So nuanced understanding of the data aided by domain awareness about the data and its limitations is crucial to building effective ML models. In general, although there is a lot of emphasis on iterating on the model, this needs to be complemented with iterations on the data itself that is fed to these models. So that's the basic conceptual idea behind a data-centric approach to ML. And although we usually think of modeling as a linear process, prepping the data first and then feeding it to a model, in practice, ML modeling needs to be circular. So model outcomes can actually loop back and inform choices about the right data to feed the model, which can add value to the entire process. So there are methods to automate this process, like identifying low quality or problematic slices in your data, which can be filtered out. These are so-called data valuation methods. So all this is, of course, known and implicitly followed. The whole ML ops framework is about it. But of late, these notions are beginning to be made more structured in practice on multiple levels. So there is a growing recent buzz around data-centric thinking, especially in vision and language applications. So Elucidata also has been advocating for this perspective for biomedical data. And you can check out our recent blog article on this topic. We have so there's also a recent article from Google Research, which basically makes the assertion that outside of internet companies and big data, most real-world ML applications are small data problems. So data quality can make a huge impact on those models. Andrew Ng, who some of you might have heard of, is a co-founder of Coursera. He has been talking and blogging quite a bit about this. And in fact, to encourage this data-centric practices, he recently also introduced this data-centric AI competition, where unlike a typical Kaggle challenge, the goal here is to improve the data so that a fixed model performs well on the given data set. So this is like one canonical example of how improving the quality of the data, removing noise in the data, can have a much larger positive impact on the performance of the model than just adding more training data. And that's taken from Andrew Ng's blog. There are also these startups now which are devoted to helping folks prepare or clean their data for using ML. So instead of focusing on the model, they focus on creating these high-value data sets. Elucidata is, of course, one player in this field. There are also other companies like Snorkel and Gretel, which basically help customers create training data sets for the ML models. And there are also now conferences being organized around data-centric themes. So in short, like, there are a lot of resources out there to explore on this perspective, and I would encourage interested folks to refer to them. So how does this data-centric approach matter for biomedical applications? So to get a better sense of this, we started to take up some case studies or projects on problems of interest to the life sciences or pharma community. And we basically try to examine the challenges around data availability and quality and the room for improving ML performance through iterations on the data. So choices around the data over and above the model itself. And how these data-centric choices can eventually improve the odds of clinical success of these models. So we have recently begun looking into this problem, predicting targeted drugs for individual patients, the whole precision approach, personalized approach applied to cancer. So in brief, in a heterogeneous disease like cancer, it is supposed that a targeted approach which factors in the genetic information about the patient can lead to safer and more effective treatment outcomes. So one example of this for a non-cancer drug is the example of genotype-guided dosing of warfarin, which is an anticoagulant for heart disease. So this drug interacts with other drugs and it's metabolized at different rates in different individuals. So recently, it was shown that use of genetic testing can help identify the correct or the safest dose for each patient. So this type of example highlights that a one-size-fits-all approach does not typically work with complex diseases, and you need a more nuanced, individual-specific treatment approach. So how is this done in practice? So as clinical data is typically not enough for training these machine learning models, what people do is to leverage in vitro data on drug sensitivity from large scale screens. So this high throughput data can be used to fit models, which basically predict how a cell line will respond to a drug, and learn useful biomarkers determining this response. And eventually this model, these models trained on cell line data need to be validated on clinical data as a first step towards coming up with clinical hypothesis, because like this XKCD cartoon sort of demonstrates, a lot of things will probably kill cancer in a petri dish, but whether that drug will work on cancer in a human without harming the human is a altogether different ballgame. So you need that additional clinical validation of a preclinical model. So this is like a more detailed schematic of this modeling process. So basically, you start by constructing a data set which is composed of data on drug responses and multi-omics profiles of cell lines. You select features, maybe by incorporating some prior biological knowledge. Then you train and evaluate models for predicting drug sensitivity. And finally, you combine these models with clinical data and validate the models on tumor samples from patients. And this could suggest interesting, ultimately, it could suggest optimal treatments for a patient or suggest opportunities for repurposing existing drugs. So as you can guess, a project like this brings together data from many different sources, data of different types. So it is important to examine the choices that are made around the data and ask how the parameters of the data, in addition to the model, can impact the outcomes of a machine learning model. So in order to motivate this, let me take a slightly reverse approach. So instead of walking you through the sequence of steps that I just showed, let's begin by looking at the performance of a model which was trained on the drug response data to predict sensitivity to a drug. So as an example, this graph shows model iterations on data for 5 drugs from a particular in vitro drug screen, GDSC 2. So for each drug, 3 different kinds of regression models were fit to the data, and the performance is compared. So as you can see, there is not much difference in the performance of the different model types. So when you replace a linear model with a random forest model, which is like typically recommended for tabular data of this size, you don't get a drastic improvement in performance. The performance remains within 10%. So this kind of result leads one to ask if there is room for improving the model performance by iterating on the data and what those data-centric choices might look like. So I'll take a few examples of such threads which look into these different data-centric angles, choices around the data, and explore the impact on the eventual accuracy of the ML models, independent of the choice of the model itself. So the first step is to take the data from this drug screen, the response data, and display it as a matrix of drugs and cell lines. So the pixelated data you see on the left side is basically different drug screens. So the data for drug responses is represented as these black and white pixels. Black pixels indicate missing values. So as you can see, there is a fair amount of missing data in these drug screens. So either those combinations of drugs and cell lines were not tested in the screen, or they did not pass the quality thresholds, and so they were not included in the final data. So given this kind of incomplete data, one may ask if it is possible to reliably fill in this missing data, and will this have any significant impact on downstream ML models. So how would one do this? So one way is to exploit correlations in the data. So basically, so the pixelated data that you see, it's not like just a bunch of random numbers. So there are patterns within the data. Like drugs can be similar in terms of what pathways they target, or cell lines can be similar in terms of what tissue or disease they come from. So there might be relationships within this data hidden in this matrix. So one could potentially exploit those correlations in the data distribution and try to guess the missing data from the available data. So we did a bit of benchmarking of some methods to impute those missing responses and compare these different methods. So basically what we did was we took a slice of this response data matrix, which was complete to begin with. We added missing values to that, so we know exactly which values are being removed. Then we applied these imputation methods to the missing data, and finally we compared the reconstructed matrix with the original matrix, and this allowed us to compare different methods in terms of like how well they can recover the original response data. So we do find that some methods do better than others, and we have picked the best method. And then we went ahead and applied that method to the actual drug response data and filled in the missing values. So we basically guessed the unavailable response data using the available response data. And the utility of this step was quantified by next comparing the models trained on the original data versus the recovered or imputed data. So this graph on the right basically shows the change in the accuracy of the models. It's called predictability score here. How that changes after we have performed data imputation. And as you can see, there is a general trend towards improved accuracy of these models after increasing the effective data set size per drug. And these differences are significant in many cases. There is, of course, some variability across these improvements, and it might differ across different screens too, as we find. But nonetheless, we feel that it's an exercise worth trying out for this kind of drug screen data. Another key data choice that one makes in this modeling process for this problem is what kind of response measure to select to use for machine learning modeling. So like if you look at the PharmacoDB database, which is available through Poly and which provides pooled data on drug responses across multiple screens. So PharmacoDB basically provides drug responses for 8 different metrics. So I mean, as you probably all know, like these drug screens basically test the viability or the growth of cell lines at different drug concentrations. All that data is pooled together to recover these dose response curves that you see on the screen. And then you can read off different metrics of drug response from this kind of a dose response curve. And PharmacoDB provides 8 different response metrics. So one obvious question is like, what is a good response metric to use? Which metric is likely to be a good proxy for like clinical drug responses? So for example, it is common to quantify drug responses in terms of IC50 values, but people have, of course, suggested that IC50 values can be unreliable when they're estimated for resistant cell lines or in case of some drugs. So it's basically like an open question to decide which metric to use. So we basically used 2 criteria to compare these different response metrics. One was how consistent is a response metric across different drug screens. So on the same pair of drug and cell line, will the response be same or very different across different screens, which might be using different experimental techniques? And the second criterion we used was, does the response metric, is it able to recover the known genetic markers of the drug from cell line data? So there are a lot of drugs like FDA approved drugs where you know exactly what kind of genetic alteration they target. So we wanted to see if these different response metrics can recover these known genetic associations from the cell line data. And so these 2 criteria might provide some kind of a idea about like the relative quality of different response metrics. So in the first exercise, we basically compared whether data from different drug screens agrees well, and how this changes across different response metrics. And what we find is that IC50 is not the best sort of response metric. There are other metrics which perform better. So the heat map on the right basically tells you the number of significant correlations between two drug screens in terms of each of those response metrics. So every column is a response metric, every row is a pair of drug screens, and every entry in that heat map basically tells you how many drugs had significantly correlated response profiles between different drug screens. So that's like a measure of consistency or robustness of that response metric. And what this suggests is that IC50 is not probably the best metric to encode drug responses. In a second experiment along those lines, what we did was to compare the so basically we asked if the drug responses can recover known genetic markers of the drug. So we essentially performed a correlation between the presence or absence of the genetic marker across all the cell lines and the profile of drug responses. And then we repeated this for every drug response metric. And then we compared these different drug responses. So the bar plot on the right basically shows the results of this comparison for 3 of those drug screens, the largest ones in PharmacoDB. And again, I mean, as you can see, IC50 does not pick out these known genetic associations so well, but there are other area above the curve and this drug sensitivity scores, which do a better job of recovering known genetic associations. So broadly, like these 2 experiments that we have tried out suggest that like IC50 may not be the best metric to use for these kinds of models, which is actually quite common in the field, and one should probably give more thought to the selection of the right response metric. Finally, as a final example of this kind of a data-centric thinking for this problem, my final example is around interpreting the performance of the trained model. So I'll show this with an example. So I picked one drug, dabrafenib. So this is an FDA-approved drug recommended for treating metastatic melanoma patients, and it targets a particular genetic mutation in the BRAF gene. So it targets the RAF pathway. So let's say, what I'm showing here is basically the result of the model. So the overall accuracy of the model is 0.62. That's the sort of correlation between the true drug responses and the predicted drug responses on the entire set of cell lines. And as you can see, the correlation is all right, pretty decent. I mean, compared to the distribution of model performance that one finds in this problem setting. But let's say we want to go ahead and apply this model only to samples from a particular disease group. So this data is for all the groups, like the entire set of cell lines. But if you say want to apply this to a particular disease group and identify sensitive from resistant cell lines. So that's sort of the kind of application setting you imagine for a precision approach, where you are interested in making distinctions between sensitive and resistant patients from a cohort drawn from a common disease, or you are interested in ordering patients for recruitment to clinical trials for a particular drug. So ideally, you would be applying such a model which was trained on all the data to a particular disease group. So if we do that, let's say we apply to skin cancer, which is where this drug happens to be recommended, we get a very different picture. So it turns out that when we evaluate the performance of the model on the skin cancer cell lines, which are shown as purple points in the scatter plot, the performance is not great. In fact, there is hardly any correlation between the predictions of the model and the true drug responses. So this suggests that the model, even though it's seems to be performing well on a global level, it doesn't do well on these smaller groups. So it seems like the model is only making these coarse distinctions between groups of cell lines. And one clue to this comes from comparing the model with a baseline model, which only used the metadata labels of these cell lines. So not the omics data, but just labels like the tissue type or the disease or experimental variables of those cell lines. And as the violin plot shows on the right, a baseline model does almost as well as the model which was trained on gene expression data. What this suggests is that the model, the expression data is not really bringing any value to predicting responses for these groups of patients. And this is happening because for this example, it turns out that there are a lot of sensitive cell lines among the skin cancer subset of the data set. So the model is probably learning to predict tissue type rather than the drug response, which it is supposed to predict. So this example suggests that global accuracy of a model might be misleading, and similar issues arise quite commonly in statistics. I mean, there is something called Simpson's paradox, which qualitative gives rise to these kinds of issues. So, I mean, given these limitations of the data, one can ask how one could probably improve the performance of this model. So, the model does well globally, like on the entire set of cell lines. It has a good test set accuracy on all the cell lines, but it doesn't do so well on the specific disease group where it needs to be applied to. So how do we improve the performance on skin cancer subsets? So we can try out a bunch of simple exercises. Let's start by seeing what happens if we use only the skin cancer cell lines to fit the model rather than using all the cell lines. However, that doesn't seem to help here. I mean, the model performance is basically around 0. So here probably there are not enough skin cancer cell lines to train a model reliably. Next, let's see how we might improve the model by pre-selecting the features that we use. So originally we used all the gene expression features to train our model, but it was probably picking up some spurious confounding factors in the data. So what if we only select features which we think might be relevant to predict drug responses for this drug? So if we pick, say, only the drug targets of the drug, or only the genes which are known to be associated with cancer, would the models give better performance? But clearly in this case, they don't. So even if we pick this pre-selected set of features, they don't seem to improve the performance dramatically. Let's try out another thing. Let's see what happens if we replace gene expression, which we have been using, with mutation data as features for the machine learning model. And as you can see, the performance of the model improves. So now it's not only performing well globally on the entire data set, but it's also performing well on these specific disease groups, skin cancer in this case. So you see that by, in this example, that by testing these different choices that we made around the data, the model outcomes can be impacted. And obviously, although we did not iterate on the model itself. So these are the sort of angles that can be explored to align the data with the problem at hand, and they can definitely add value to the modeling process. So this is like the sort of where we started and where we got to. So we started with a model which performed well globally, but did not perform well on skin cancer cell lines, and then we basically made these choices around the data and the features. And then we got an improvement in this subgroup performance of the model also. So this sort of brings me to the end of my presentation. We looked at a specific example of how data-centric angles might apply to an ML project using biomolecular omics data. My intent here was not to present clean, complete results, but rather to motivate the role of iterations on the data as a complement to the model-centric approach, which is typically the norm. And iterations on the data are an essential part of an MLOps process. In fact, as we saw with the examples that I talked about, data might actually make a bigger impact on the ML outcomes than tweaking the model itself. So I gave a glimpse of some of those data-centric angles. Some of them are, of course, generic. Some are specific to the problem context. There are other threads apart from the ones I brought up, which, of course, I didn't have time to go through, but which we are pursuing at LUC Data. So as a data and ML ops organization, we have been advocating for the value of data quality for ML projects. And basically, I tried to convey a sense of that through this ongoing case study. So I would like to thank you for your attention. If you have any questions, you can write to me or feel free to connect with us on social media, which I'm just reiterating what Kritika said. I'll end by thanking my colleagues at LUC Data who have been involved in this project and also helped with the content. And of course, please watch out for our upcoming Data Fair virtual event in October to know more about our project. So thanks for your time.
Thank you so much, sir, for an amazing talk. We have one question in the chat box. What percentage of missing data is acceptable for imputation? Does imputation leads to overfitting?
Yeah, so that's a good question. So that's something something that's like work in progress for us. So one thing we do find that there is difference in the quality of imputation across different drug screens, which might have different quantities of missing data. It can also depend on how the missing data is distributed. So whether it is like these missing values are randomly distributed across different drugs, or are they clustered together within certain drugs. So that is sort of an ongoing exercise for us to figure out how much data can be missing and you can still recover it. So there is, of course, a trade-off. So if you have too much of missing data, you won't be able to guess the missing data because there is little information to do that. On the other hand, if you have very little missing data, then imputation won't add any value because it's not really going to make a big impact. So there is probably a sweet spot where like data imputation actually helps, and we feel that depends on the data, like the drug screen that you're looking at. So clearly, like it's hard to answer that question in general, but it's definitely worth applying it to your drug screen.
I hope Sati got her answer. And if anybody else has any question, they can post in the chat box.
Yes, Sati says thank you. And thank you so much, Dr. Gaurang, for your excellent presentation. It was indeed a very nice presentation to understand everything about the functionalities and the basics around it. So we thank you immensely for joining us and giving the presentation and talking about the area.
Thank you. Thank you for the opportunity.
Thank you so much. And we look forward to more such sessions in future.
Thank you.
Thank you.
Good luck to everyone.
Thank you so much. Okay, so I believe we are waiting for our next speaker. He's yet to join us, I think. Is it so, Payal? Can you see?
Yes, ma'am. He has not joined yet.
Mr. Shashank Jatav is not there. I think we'll take a short break till the time Mr. Shashank joins, and then we can continue the session.
Sure. So we'll have a short break for 10 minutes. I request everyone to join back at.
Please stay online, and you can take a short break and come back. So we will start as soon as Mr. Shashank joins.
Welcome back, everyone. Our session is on biomedical data curation through data-centric AI. I'm Mr. Shashank Jatav, LUC Data, New Delhi. Mr. Shashank joined LUC Data in 2016 as a senior data scientist and is currently working as the director of data products. He received his bachelor's in mechanical engineering at IIT Kanpur, and his main skills include coding. I now request Mr. Shashank Jatav to start the session.
Hello, everyone. I'll just start the screen share. Just give me a sec. Are you able to see my screen?
Yes.
Visible.
Okay, so I'm hoping like everyone knows about AI or machine learning. If not, then I will like just ask. And also like if you want to ask any questions in between, feel free to ask any of the questions as we go. So I mean, if I just don't wait for me to finish the presentation. You can ask it anytime. So basically, so for any AI system to work, there are two most important things, and we were like, I mean, these are very common sensible things like the data as well as the model. I don't know if anyone has used phones, for example, if anyone has an iPhone or any like Google Photos, for example, if you see, right? So the Google Photos, they detect your face, and they also detect your um, they also detect different persons and also like ask you to tag them, um, and they can also make memories of that person, and so that's an example of an AI system. Um, and the data that on which they are trained is basically I mean, Apple and Google, they have vast amounts of um, image data to train how a human face looks like, and the model on which they are trained have it's a neural network based architecture. So basically they have uh, I don't want to get into details of that system, but CNN uh, and transformers based models which can basically get it. So model based basically in the brief history of AI that is there, advances in model have been by leap and bounds. What I mean by that is like if you know about ImageNet, and basically ImageNet is one such kind of a data set on which people train their models. Um, Models for recognizing any any kind of face, or there are like 1,000 different entities in there. So that's the data set that people used to, or they are still like basically try to surpass human accuracy on that. And so that is the data part of it. The second part is basically the model, and people have been trying out different architectures or different models for. Just a sec, okay, someone is sorry. Different architectures for or different models for that kind of data, and there have been a history of improvement of those models, and now those models are reaching human accuracy. Um, and a big part of that is also that ImageNet is also basically cleaned up, and there are errors in the ImageNet that people have to basically make sure that they are not present, human errors, and also like um, ImageNet is also based on basically they have done this curation through Amazon Turk, which is a AWS based. Basically, humans have done it, but there are chances of a lot of errors in that. So basically, the model based accuracy has basically increased by leap and bounds. But if we have to increase the accuracy any further, we have to work on data. We have to make sure that the data is all right. The data is not biased, and the data is basically of good quality. So both of these, I mean, the data and model both are very important for an AI system. And model is something that I mean, like a lot of folks in the world are not bothered about. They just basically use any out of the box model to get started. And in recent scenario, people just have to work on data. So data now is becoming the focus, right? So yeah, so both are important, but the AI community has focused a lot on improving and iterating the model on different benchmarks. Um, and they have been generally successful at that, but very few have been trying to do it on the quality of the data. So, and that is true for natural language processing. So at Illustrator, we generally use a lot of NLP techniques to basically curate data. And we don't try to innovate on the model, but we try to innovate on the data. How do we get more and more data? There is this recent paper, I think it came this week itself by OpenAI, and you should search for it. Whisper, there's a model called Whisper, which is a huge, they did not change any model, but they basically used a very big corpus. By corpus, I mean like the data set to basically train the model. And the way they did it was completely using weak supervision and database approaches. So that also tells you that even big models, companies which are basically innovating on models are moving towards data in order to basically get more state-of-the-art models. So that's brief history about models and data. So what is language model and what is NER? So if you see on the slide, then basically language model is not something very new. It has been there for a long time. You must have heard of, or if anyone has done language modeling, they must have heard of hidden Markov models and some probabilistic models to basically model language. So what does modeling language simply means is that like if I have like some 5 words, then I have to predict the next word. For example, you can see that here itself, autophagy maintains tumor growth through circulating dash. What are these basically? What can be here is something that the model has to predict. And this was earlier basically done for normal language, but not for biomedical text. But now people have done it for biomedical text through models like BioBert, PubMedBert, etc. So anything which is colored here is actually NER, which is called a named entity recognition. So that's the task that we work on a lot. And you can see tumor is a disease, starvation intolerance is another disease, arginine is a drug. So you identify what is there what is written in the paragraph, and where are the entities. So that's basically named entity recognition. And that we do a lot in order to make the data fair. So how do we do training a language model? We basically we have this text data with labels, and that we generate in-house. We have a lot of curators who actually mark these text that where is this particular named entity coming in. And you have a language model, and then you have a loss function, and then you train the model to learn this particular task. And at the inference time, basically, you spit out the label. So as simple as that, right? So basically, I told you about BERT and BioBERT earlier. Feel free to look it up. Now, BERT is basically an architecture which and with some predefined tasks, with some basically with some multi-head attention models. Now, I mean, these are jargons in BERT, but all that is happening is basically it has a very big neural network. It has around like 110 million weights that are present in BERT. And BioBERT is trained on medical corpus. By corpus, I mean on medical text, while BERT is trained on Wikipedia and general text. So BERT does not perform very well on biomedical data, while BioBERT does. So that this has been proven. And BioBERT basically does a good job of basically recognizing entities or any other kind of biomedical task that you throw at it. I mean, what so our current model development process, and why am I showing you this, is basically why you want to focus on data. What does it lead to? So one of the way to look at it is basically the current model development process has a like 2 week of manual curation process, which we do, and it has a model tuning experimental process that we do as well. And this is not data centric because we do all of these manual curation manually. So this takes about 6 weeks to develop a model for a particular field, which is a bit too long. So so basically manual curation training takes time, and that is what basically these data centric approaches help you overcome. Now, how do they help you overcome? It's basically shortening the time for manual curation and also the model development process. So, I mean, I don't know if you folks know about supervised training. So whatever I showed you earlier was all supervised training of models. So weak supervision is basically another way to do it is, and this is what the Whisper paper from OpenAI also did very recently. The paper came from this week itself. Now the weak supervision has a problem that you don't have any label data, right? Now, if you don't have any label data, then how do you train it? So what you do is like you talk to some of the domain experts and you make certain rules for recognizing entities, and you make a lot of such rules, and then you basically train your BERT model on that weakly supervised data. Now, what it does is it basically the BERT is good at removing false positives from this weak supervision. That is what we have seen. And what it does is basically it reduces the time of manual curation that earlier we had to do like text by text, one at a time, to basically just making these rules, and those rules can be used for labeling the data. And that is much faster than you can do it within 2 or 3 days as compared to 2 or 3 weeks for manual curation. And what's active learning? So active learning is basically what you do is you try to learn as you go, in the sense like while you are basically labeling the data, you train the model itself, and you have a golden corpus already ready, golden data set already ready, and you basically you do the model training as you are labeling the data, and you pick the harder examples first. What does that mean is basically if for example you can recognize acute myeloid leukemia as being a disease, but like if there is a harder context for a particular sample, like this person was diagnosed with AML or leukemia, those might be like the acute myeloid leukemia is not there and you're only dealing with leukemia. That might be a harder thing to resolve for manual curators. So we rank these. Rank these basically labels and do the harder things first, and that is what is basically the concept of active learning. So active learning combined with weak supervision basically shortens the time of labeling the data. So that's what we mean by data-centric approaches, where you are trying to improve the data faster rather than iterating on the model. The model is already very well researched. BERT models and BERT architectures can learn almost anything. That is what we have seen. So this is what I was explaining earlier, where you have a domain expert. They basically have these labeling functions, and then you basically add it to a label model. And this label model can be a very a simple model, as to like basically you will eliminate what it does is basically is try to see what is the overlap between two labeling functions, and tries to see if different labeling functions are marking the same thing, and it tries to make a model on that. And then you basically it can be a hidden Markov model or something like that. And then you finally make a discriminative model, which is more of a probabilistic model, which is a BERT model per se. So this is weak supervision. And yeah, so this is what I was telling earlier, that as you can see on the graph, that if you do only weak supervision, then you might have a lot of false positives. Like here, in this case, you can see there are like 179 false positives here. But after training with BioBird, the false positives decrease a lot. And that is the power of, these deep learning models, where basically they can identify false positives easily and improve the performance. So yeah, I mean, the manual, if you train a model with only manual curation, of course, you would have a better accuracy as compared to a weak supervision model. But you can quickly basically eliminate that using active learning. So there, basically, you can put weak supervision model as part of you can put a weak supervision model as part of an active learning module, and the model will tell curators as to like what it thinks is the right answer, and all curators will have to say is like whether it is right or wrong, or if it is wrong, then what else it is. So it also makes the job of manual curators easy as well. So that's basically active learning. And I mean, this is how it kind of works. It basically, you have this model, which is, you can say, our weak supervision model, and you have this unlabeled data set. You label it up using this learning model, and then basically you take on all the harder, or basically the models, the labels which are wrong, and you ask manual curators to curate only those. It is not easy to get like labels which are again wrong, because again, if you have to do that, then you have to label it from scratch, which is again a problem, right? So what you do is like you take the model's confidence, and you only focus on those labels where the confidence of the model is lower. So this is active learning. Do you have any questions till now? Anyone has any questions? Okay, so so this was active learning, and why do we use active learning is basically if you just use random sampling, for example, and you don't use models prediction, confidence score, then basically you'll have to label a lot more rather than lesser stuff. So basically you'll have to label double, as you can see from this graph, double or more. while in active learning, you have basically random sampling needed around 3000 to reach the same accuracy level as compared to active learning, which took around only 1200. So that's the benefit of active learning. And if you combine weak supervision and active learning, what can you say your question, whoever it is? Are they allowed to speak? Himanshu, right? Can you unmute yourself and ask the question if possible?
They're actually just attending as attendees, so I don't think they can unmute themselves. They can only communicate through the chat box.
Okay. So I don't understand the question, but I'm guessing that you're asking how to do error analysis in a model. So basically, if the error analysis is kind of an art to do. For deep learning models, the error analysis is very hard to do. So, I mean, for deep learning models, you would have to focus on data itself. And there is this very good paper by by some authors who have actually tried to do error analysis on deep learning models for chest X-ray. So what they did was they divided the data into different parts and then try to see like why a model is doing something what it is doing. So in deep learning, the only way to figure that out is through like basically look at the data where it is making errors and then try to identify what it might be. So it's a very blinded approach. And if you are not using deep learning models, you're using traditional machine learning, then you have more options. For example, if you are using random forest model, then you can use SHAP to determine which feature is basically causing an error. And that will help you out. If you're using simple logistic regression, then you can simply figure out as to plot the decision boundary and figure out what is the where model is making an error and why it is so. So I mean, traditional machine learning, it is easier to do it. In deep learning, you have to just look only at the data. And once you do the error analysis, you try to remove those errors from the data, or you use a better model. In case of traditional machine learning, there are a lot of different things that you can tweak, like regularization. Depends on whether you think it is underfitting or overfitting the data. So, I mean, it's a very broad question. The answer to that is two error analysis, and the error analysis method for doing error analysis is different for different kind of models. So you need to know what kind of model you are looking to and how you can apply or how you can do error analysis in it. Does that make sense, Himanshu? Okay. Yeah, so coming back to weak supervision and active learning. So basically you can see that the weak supervision plus active model, even though it use a little bit more manual labels than only active learning, but it reached a higher accuracy. And basically that is something, and also like weak supervision is faster than just manual labels. So this will be faster than others. So that is what basically these two, I mean, in data-centric NLP, there are other techniques as well, but I'm just focusing on these two for now. Yeah, so earlier when we talked about the duration of this whole process, so it was taking about 6 weeks to complete it. But with the data-centric approaches, we were able to reduce it to 4 weeks or lesser. So yeah, so basically this this is how basically we were able to do that. I think I'm done with the talk.
So we have one question, I think, in the chat box.
Yeah, so there are some tools available. I mean, people are building those tools for interpretation of black box models, for example. Um, you should search Weight Watcher, which is one tool. But as you know, in deep learning, basically, you don't you don't do any feature extraction, right? Or you don't select any features. So the problem with that is if you don't know what features you are selecting, then you don't know what is it that the model is working on, right? So for black box models, there's only like two ways to do it. I mean, again, model or on data focused. For model, you can use tools like Weight Watcher, etc., to see if your model is overfitting or underfitting. Just search Weight Watcher. Um, um, that is one way to look at basically, and you can also use, in case of BERT, you can use BERT Viz to understand how different weights are contributing to a particular task. So you can do that much, but apart from that, you can do nothing much. The only thing that you can do is like basically you can look at the data itself to see how model is why model is doing what it is doing. As I said earlier, there is this paper about chest X-ray. So what authors there did was they did not made any they did not make any assumptions or interpretation about the model itself. What they did was they divided the data into three categories, and out of those three categories, they basically made a sharp metric which you can use or anyone can use to interpret those uh, imaging model output, right? So again, that was completely based on data. Like basically, they were just looking at why data and human interpretation, why a particular X-ray was being diagnosed as something while the others were not, right? So short answer is in DL or the black box models, there is no silver bullet. There are no concrete tools available right now to basically interpret the model, but you can derive some sort of actionable insight by looking at data or looking at the weights. That is still possible, but yeah. Any other questions?
I hope Siddharth got his answer. If anybody else has any question, they can post it in the chat box. I don't think there is any other question. Thank you so much, sir, for such an interesting talk.
Yeah, thank you for having me. Okay,
We'll have a short break. We'll have a short break. Please stay online. We'll start the session as soon as the next speaker joins. Thank you, everyone.
I believe our next speaker is not there, Payal.
So hi, welcome back everyone. Our next session is on querying heterogeneous biomedical data on Polypublic Omics Atlas by Ms. Anaga Ashok, Elucidata, New Delhi. Anaga Ashok pursued her BTech in biotechnology from TES University, Bangalore, and a PG diploma in big data biology from IDAB, Bangalore. She's currently working as a data analyst at Elucidata. I now request Ms. Anaga Ashok to please start the session.
Yeah, thank you so much for the introduction. And before starting this session, I'd like to know more about the people who are attending this session, just so I know how I can move forward. So you guys can raise your hand, or if there's any other reaction that you can give.
They can put their questions in the chat box or interact through the chat box with you.
Oh, okay. So just type yes in the chat box. I just wanted to know how many of you are familiar with coding. Okay, I see that 3 people have responded. So I'm guessing that most of you are not that familiar with coding. Uh, so another thing I wanted to know is how many of you are familiar with sequencing data? Any type of sequencing data, and it could be conceptual information as well.
I think most of our participants would be familiar with sequencing data. Not all of them might have had hands-on experience with coding, but then since they are mostly from biotechnology field, they will be familiar with the sequencing data.
Okay, that's amazing. So let's move on to the session. Let me share my screen. So hi everyone, I'm Anagha, as uh, someone introduced me before, and today I will be conducting the session on heterogeneous data querying on Poly. Um, Before going on to the hands-on session, I'd like to offer some background information on it. If you have any questions, please post them in the chat. And before going to the hands-on session, I can take a look at the chat and answer some of your questions. So coming to what biomedical data is. So biomedical data is data that's generated in biological experiments, such as RNA-seq data, DNA data. And we can see in this diagram that the biomedical data that is generated over years has increased by a lot. So there's a lot of difference in data that was generated in the year, say 1990 to the year 2020. And this is because there has been a lot of improvement in our sequencing technology, because of which there's a lot of data that's being generated today. The sequencing technology has been made more accessible, it has been made more efficient, it's made more cost effective, and that's the reason it's being used a lot more than before today. So with a lot of biomedical data that we have, there's a lot of this data which is publicly available as well. But just because this data is publicly available, it doesn't mean it's usable. And that is because the data is not fair, which corresponds to data being findable, accessible, interoperable, and usable. So this might be because at public sources, the data might be in different file formats. So one experiment might generate data uh, in, say, a TXT file, another might generate it in CSV file, and the way they organize data might also be different. They might, some experiments in the public repositories might have raw data, some might have processed data, and there's a lot of inconsistencies in pipelines that are used to process data as well, which makes it difficult to use. And in public data, there are a lot of repositories, and to find multiple data types, so suppose there's a repository for transcriptomics, another for proteomics, you will have to be familiar with the search engines of both platforms in order to find data sets that you want. And in case of proprietary data, it has low interoperability and reusability, because it might happen that the data engineering and data science skills are limited in the proprietary data case. So in order to, so let's move on to what happens in multi-omics research today. So there's a lot of different kinds of data that's available today, which is proteomics, metabolomics, transcriptomics. The figure here shows different different aspects or different molecules that we can sequence and generate data from. And some of the main steps that are involved in multi-omics research is to go through dozens of repositories or databases, downloading the data sets that we want, cleaning them up, and preparing them for the analysis, harmonization of metadata. Metadata is nothing but information about your experiment. So suppose you have a data set, a transcriptomics data set, which has 3 samples and it has measured gene expression in those 3 samples. So the metadata for this experiment could be the information about the samples, like where they come from, which patient they are from, what is the tissue type. And there could be metadata about the genes itself, what is the location of the genes, what are the types, or if the genes are membrane bound or not. So there can be these levels of metadata, and There can be metadata about the data set itself, like what the experiment is based on, what were the samples that were chosen, how many samples the data set has, why were they chosen, what is the disease of this, what is the disease that this experiment studies, all of that. And then comes the analysis part. Then there is communication and sharing, updating workflows, maintaining pipelines, and repeating the steps above. So here we can see that there are over 4 to 5 steps before we actually do the analysis. And these are the steps that take most time when we analyze a data or when we work on biomedical data analysis. And sometimes it might just so happen that 80% of your time goes on these steps and analysis just takes 20% of your time. So in order to save time and move on and provide data that is ready for analysis, we have something called a public omics atlas, which is harmonized ML-ready searchable collection of data sets on Poly. And what a omics atlas is that it contains public data sets from different sources. So there are different public sources like Geo, Links, CBioportal, TCGA. The data is taken from these sources and they are passed through two main steps, which is data engineering and metadata harmonization. And then the data is structured, is brought into consistent file formats, and then it's stored on what we call an omics atlas. An omics atlas is nothing but a poly repository that holds these structured data sets. And these data sets can be accessed by using a programmatic way, which is through PolyPython, which is a Python library that we have so that you can connect to the omics atlas and use these data sets. Next is you can also access these data sets through a user interface. And we also have app integrations with omics atlas. So we have third-party applications such as cell by gene to visualize single cell data. And we also have some internal tools that are generated, and there is integration with tools like Spotfire as well, which are used for dashboarding. So here in this figure, as we can see, there are more than 1.6 million data sets, 26 different data types from 30 different sources. So you can imagine how easy this would make multi-omics research, because you get different data types from 30 different data sources on one platform. You need not be familiar with, like, say, if you are using 5 data types for your multi-omics research, and if you have public databases that are very specific to these 5 data types, you need to be familiar with all of these 5 platforms, find the data sets which are not organized on that platform, by the way, and then clean it and then feed it into your analysis. You can have that all over here, which is structured data, multiple sources at one platform. So that's the public omics atlas. And for those who are not familiar with any of the public data resources, I'll go through 2 very popular data resources that researchers used in their experiments very commonly. So the first one is NCBI GEO, which stands for Gene Expression Omnibus. It has different types of data, that is microarray data, next generation sequencing data such as RNA-seq, genome data, and other high throughput functional data. And a GEO data set is a collection of comparable samples, which is processed using the same platform. And the GEO repository has variety of data or experiments. So it might have experiments that is based on patients. It might have experiments which is based on cell lines. It might have experiments from cell cultures. It might have experiments from animals. So there's a lot of variety in GEO, and this is one of the most popular repositories among researchers. The next one is TCGA, which is the Cancer Genome Atlas. So the Cancer Genome Atlas concentrates on genomic data from genomic data in cancer, and their aim is to understand molecular basis of cancer using this genomic data and analyzing this genomic data. So these are the 2 data sources that are majorly popular. And TCGA also has different data types. such as copy number variation, proteomics, transcriptomics. So samples from different patients are taken and they are sequenced to get the data for each of these samples. Moving on to the file formats that we have on Poly, we have the GCT file format, which is commonly used for most of the data types on our platform. And this file format stores both the metadata and the data in one file. So here we can see that there is the red part is the measurement, or suppose again this is a transcriptomics data set. PSME1, CISD1, they are all genes. And suppose this experiment has 4 samples, sample 1, sample 2, sample 3, and 4. So 9.345 is the expression value for PSME1 in sample 1, and similarly the other values. And here we can see that there is metadata information for samples as well. So there is information for sample 1, which is like maybe it's treated with a DMSO for some amount of time. This is treated with another drug. Sample 3 is treated with another drug, and there is metadata for the genes as well. And these might be the gene locations. So this is the GCT file format. And next is the H5AD file format, which is the file format we use for single cell data. So single cell data is slightly different from the other data types. In single cell data, we sequence the expression values from each cell. So imagine you have one sample and you're breaking it down into individual cells and sequencing those individual cells. So we can imagine that there's a huge amount of data that is generated by the end of it. So to store this huge amount of data, we need a more efficient file format, and that is the H5AD file format. So this has a data matrix of the shape, the number of cells into the number of genes. And then we have a data matrix or a data frame for annotation of the variables or the features or the genes, basically entities that are measured. And OBS, which is observation, which is the annotation of the observation or the cells. So this might have information like cell type or information of the disease status, information of tissue. And then there is other information that's stored about the matrix as well, which is here UNS is unstructured annotation, where the metadata is stored in a dictionary format or in a key value pair. So I hope this is clear. And next is, why do multi-omics data analysis in the first place? So starting with what is multi-omics data? It is data from different omes, such as genome, proteome, transcriptome, metabolome. And understanding of the molecular basis is way better when you have multi-omics data, rather than just sticking to transcriptomics data or metabolomics data. And this will help us connect the genotype better to the phenotype, and this can be used to discover novel drugs or to discover very minute mechanisms that play a major role, such as discovering mechanisms for drug resistance in cancer. So this is where multi-omics data comes in to be useful. Yeah, so before moving to this part, like I said, I'll just view the questions in the chat.
There is one
How
question.
is Omics Atlas different from available biomedical data repository? Let's take Geo as an example in this case. So if you go to Geo and search for studies, you can get a lot of studies on Geo, but these are very unstructured studies. So one thing is that you will need to read through the description of those studies. You will need to read through the publication to understand what those studies are based on, whether those studies are patient studies, whether those studies are animal studies, if your experiment is very specific to it. Next is, when we download files from Geo, they might be in different file formats. So suppose you want at least 10 data sets for experimental analysis, but all of the 10 data sets that you find from Geo have a different file format. So say one experiment stores it in TXT file format, the other stores it in CSV, the other stores it in a JSON file, or something like that. So the file formats are very different, and the way data is organized is also different. So by organized, I mean there's a data matrix, and there's information about the samples. So capturing the sample information, mapping it to the data, all of this takes a lot of time. And that is what we work on. We make the metadata and data, we connect the metadata and data on our platform, and we structure it, we provide it in the same file format, consistent file formats, not only within a repository, but across different repositories. And that is a major advantage that Omics Atlas offers over publicly available data repositories that store data. Does that answer your question? Cool. So let's move on to the hands-on session. So first, we'll need to log in to the Poly platform. I'll first go through the UI and then I'll go through the code. Make sure that you concentrate on this part because you'll be using a lot of this in your next hands-on sessions as well. So let me send this in. So this is the login page. Everyone, please go through. So I'll give it a minute or a few seconds until everyone are on this page. So once you're on the page, you will need to log in. So your login is amity.workshop at lucdata.io, and your password is amity-sept 25 star. So let me just post this in the chat as well before logging in. Yeah, so I've posted the email and the password. Oh, the password is slightly wrong. The A is capital, by the way. Let me just type it again. Yeah, the second password is the correct one. The A is capital in the password. So once you have typed it out, press on login, and that will open up the Poly account. So everyone has been provided with the same Poly account and will be working in the same account. So as you log in, I'm thinking that this is the page that is displayed in your case. Please react with a yes if you have already logged in. I hope everyone has logged in by now. Any one of you can type yes, and I'll move on. Oh, cool. So this is the home page of Poly, and we can see that there's a number of data sets that's shown, data sources, and 26 different data types. And on the left side, you can see that there are a bunch of options. So there's an option for home, which is this page. There's an option for omics atlases. Next, there's the option for workspace, for applications, for curation. And then there are some user options at the bottom and notification. And here you have the documentation as well. So if you click on it, it opens the documentation. You can go through the documentation to read about Poly in detail. But for now, in the practical session, let's see how an omics atlas looks on the UI. So click on the omics atlases option and it will open up the omics atlas page. And here in all omics atlas, you can see that there are two options, mock therapeutics and import. So these are the two omics atlas that are linked with these accounts. In this session, I'll be using mock therapeutics. In the next session, import omics atlas will be covered. So let's click on mock therapeutics. And here we can see that there's a summary of the omics atlas given. We can see that there is number of tissues, diseases, organisms, and cell lines that are listed out here. And we have charts to show the distribution of some of the categories. So this is a bar chart showing how many data sets are from import, geo, the different data types that are available, different organisms, and how many data sets are there for each organism, different diseases, and how many data sets are there for those diseases. So let's click on view all data button to see the data sets. So click on it and it will show you, or it will list out all of the data sets that are there in the omics atlas. So at the left side, you can see that there are filters available to filter out the data set. There is a filter for sample where you can specify a range saying that I only want experiments with samples that are more than this and this number. So if you want an experiment with more than 50 samples, if you want an experiment more than 100 samples, you can provide 100 as the minimum over here, click on go, and it will filter out the data sets accordingly. Similarly, you have filters for drugs, you have filters for tissue, data type, disease. So to filter out these data sets and to show you how easy it is to find data sets that you want, let's consider Alzheimer's disease because that's what I've used in the code as well. So select Alzheimer's disease and disease. If you don't find the disease that you are searching for, if you're searching for another instance, you can just type in it saying Parkinson's and it'll give you Parkinson's disease. So I've already chosen Alzheimer's disease over here, and suppose I want it to be on a tissue for cerebral cortex. So you can select cerebral cortex in the tissue, and then I only want data sets for the transcriptomics data type. So under the data type filter option, you can select transcriptomics, and this leaves us with this data set. So this is the data set that I can carry on to my analysis. So this is how easy it is to find data set on Poly. You have all of these columns, and this is enabled because the data has been extremely structured before being incorporated on Poly. So similarly, if you want a particular data type, suppose for your experiment, you want both proteomics and drug response, you can just select these two options and it gives you all of the data sets for proteomics and drug response. You need not go anywhere else to a different repository where these data types are available and then search for these data sets again. So I'll just pause and see if there are any questions that people have posted.
I don't think there are any
I see no
questions.
questions on the chat. So I hope all of you have taken a look at the omics atlas. So if you have, just type a yes so that I can move on to the next part, which is the code. Is everyone clear with how to access data sets? Okay. So this is the part for filtering the data sets. In order to view details about the data sets, that can be done on the UI as well. So let's just go back to the filter that we had provided. And if I click on view details button, which is at the right side of a data set, it will display the sample distribution, and it'll display an overall summary of the data set itself. So you can see that there are different charts that are there to explain what is in the data set and what is the distribution of samples in the data set. So these are charts, and if you want to see the information itself, you can click on this option next to samples, and that will be available. And here you can see that there's a description, there's number of samples, and there's a link for source. So this redirects you to the page on Geo where this data set is stored. So if I click on it, it just opens up the Geo page for this data set. And next we can see that there are different tags. So these are tags of disease, tissue, data type that is allotted to this data set so that it's easy to find this data set.
So we actually have a message on the chat box.
Okay.
Debriti says, if I filter it against proteomics and drug response and seeing a page can't find data set, did I miss something? Please explain once more.
Oh, the reason you're seeing it, I'm assuming that you filtered for Alzheimer's transcriptomics and you have further provided a filter for drug response and proteomics. And that's because in this omics atlas, there is no Alzheimer's data set, which is proteomics, which is of the data type proteomics and drug response. This is a omics atlas that is prepared for this workshop, so it has limited number of data sets. So if I were to go on the actual geo data sets, we would find it. Okay. So moving on to the next part, which is the code. So before understanding the code, let's go back and learn more about the Poly platform itself. So in these 4 options, let's go to the workspace option. So on Poly, workspace is like a folder where you can create. So here you can create different workspaces. It's equivalent to creating different folders. And within that workspace, you can store your files. So it could be an analysis. Analysis is nothing but a state of the apps that we host. And that is the state that is saved. So suppose you run Cell by Gene app and you generate some visualizations. You can save that state and that will be saved as the analysis type. So when you load it, you will see the same visualizations that you had generated before. And similarly, you have notebooks, which is Poly notebooks, and you can store some other file types as well, such as CSV files and H5AD files. So here, for your hands-on workshop, you will need to create your own workspace. So to create your own workspace, please click on New Workspace button and provide a name for the workspace. You could provide your name plus your roll number and provide a description for the workspace, like Amity workshop, hands-on session, something like that, and click on save. So I'll create my own workspace, which is Anagha LOC data, and a description of Amity hands-on workshop. So when you click on save, it creates this new workspace. So here at the left side, we can see that Anagha LOC data has been created. So I'll give a minute to you guys and create your own workspaces so that we can move on. Okay, let's move to the next part. And there is this workspace for Amity Uni workshop. So please go there. And here there are all the demo notebooks that we will be using in this session. So the notebook that I will be using for my session is the heterogeneous data workshop.ipynb. For those who are not familiar with what a notebook is, this is a Poly notebook and it's very familiar to a Jupyter notebook. A Jupyter notebook is an application in which you can have cells where there is a section for writing your code. There's a section where your output is displayed, and there is a section where you can provide description of the code. And in Poly notebook, you can shift through 3 different languages, which is Bash, Python, and R. And that is useful because within one notebook or within one document as such, you can have different languages and you can have different analysis, or you can run different functions in different languages in one environment or in one place. So so what we need to do now is we need to move this notebook into the workspace that you have created. So select this notebook, click on it, and it'll display a few things on the right side. So there is details, there's versions, and there is 4 options above, which is for preview, copy, move, and delete. So if you click on preview, it just shows you what is within the notebook. So here it just shows you what is there in the notebook. It does not really, you can't really execute this code or do anything with it. This is just for viewing what is there. Okay, so someone has already copied it. So yeah, and then there is also this README file. So if there are people who are joining this workshop later on, please tell them to refer this file. You can just click on view, and there are steps on how you can create a workspace, how you can copy a notebook, and how you can open a notebook. So yeah, to copy this notebook, you can press on copy, select your workspace name, which is Anagha LOC data for me, and click on copy file.
So the link is not working for one of the participants there. That on the chat box.
Unable to log into the link given above. Can you just type LOC data in Google and go to their main page? So just type LOC data, go to this page, and click on login over here at the top right. it'll show the page. And then use the credentials. And I think you might be using the password that I've given before, which is the Amity except with small a. Use the password with the capital A. Okay, moving on. I think if you have more technical issues, you can just put it up in the chat. And my colleague Shraddha is also there in this meet, so she can respond to you. So moving on, I hope that each of your workspaces has the notebook. Now to open up the notebook, you can just click on it and there will be a button called Edit and Launch. So click on it and it will give you a few options. So here you need to select the Docker environment. What a Docker environment is, it's just a predefined, it's just a pre-configured environment, which can be useful to your analysis. So here we have different environments for proteomics. We have different environments for RNA-seq, single cell. For this session, it's not really based on any analysis or anything. We are just going to be querying data sets from Omics Atlas. So you can just choose Python 3.10 and it does not, and here you have options for machine type as well. So this is the computation, this allows you to choose the computation power that you require for your analysis. Some analysis require a lot of high computation power and some need not require a lot of it. So for this session, we can choose the poly small, which is the 2 GB RAM option, because again, this session does not have any analysis and it's not that computationally intensive. So once you choose these two options, click on launch and it will open up a new tab in which the notebook will open. So right now it's loading. So the notebook is starting up and once this completely loads, your notebook will open. Okay, now that your notebook is open, this is what a poly notebook looks like. So here where it's written, welcome to Poly Python 3 notebook, this is the descriptive space or cell. And then this cell in which there is sudo pip3 install polypython, that is the cell for code. And below there is a section for displaying the output. So if everyone has, I hope everyone has managed to open this notebook. I'll give you some few seconds and then we'll start going through the notebook. Okay, I hope the notebook has opened up for everyone. Let's start going through it. So the first step is to install the polypython module in the notebook. So you can execute this cell by selecting it and clicking on cell and selecting the run cells option, or you can just click on it and click on this option, which is run, which is next to the stop option. If I click on it, it'll run the cell, or you can select the cell and press shift enter on your keyboard and that will run the cell as well. So I hope everyone is able to run the cells in the Jupyter notebook. So after installing this module, we can import the libraries or we can import the modules that are required. For those who are not familiar with programming at all, modules are like pre-written code that you can get in your environment or in your notebook, so that you can use them to for your analysis, or you can use these functions in your notebook to perform the function. Or so that is what a library or a module is. It's just a set of pre-written functions that you can use in your code. So you need not type a huge amount of code and for each function. You can just have these pre-written functions and you can just run them instead. So after importing these modules, you have to authorize and connect to Poly Omics Atlas. So here, this is the code which fetches the auth token or the key through which you can connect to Poly. And this is the code to connect to Poly, where we are just saying Poly auth and auth token. And then we are defining a Omics Atlas object and connecting to Omics Atlas by specifying, by connecting to the class Omics Atlas that we just imported. So you can go ahead and run that cell. Oh, by the way, once how you determine that the cell has already run is at the left side here beside in bracket 4. When you're running the cell, there'll be a star or an asterisk, and when the code is finished running, there'll be a number over there. So yeah, this is the authorizing. You can also find this auth token on UI. So if you go back and go to user options and authentication, you have your authentication token as well. So you can copy it from here as well, but this code just automatically fetches it. So it's more convenient. And there is PD set option. This is just for display purposes. Next is getting omics atlas summary through code. So here we can execute a function named omics atlas underscore summary, which is included in the class omics atlas, which is in the object omics atlas that we just defined. So to get the summary, what we need to do is omics atlas dot omics atlas underscore summary and provide the OA name or the omics atlas name, which is mock therapeutics. And then this part, which is pd dot data frame from dict, this is just for visualizing the data that is fetched by this function. This just makes the visualization easier. So run the cell and it will display the summary. So here you can see that there's count of cell line, cell type, the summary of the omics atlas, like an overall picture of what is in the omics atlas is shown. Now coming to the metadata fields on Poly, like I said, there is different types of metadata that can be associated with the data set. There can be data set level and sample level. So we have metadata fields at two levels, which is data set level, where the metadata just depicts a general view of what the data set is. And for sample level, it stores the information about disease for each sample or subtype for each sample, gender, characteristics, other information for each sample. So let's see what tables are there in the omics atlas. These tables store the metadata. To view the tables or the entities that store metadata in the omics atlas, you need to run a SQL query called which goes like this, which is show tables in mock therapeutics. And you run the query using the query underscore metadata function in omics atlas, pass it the query that you have typed over here, and then view the results. So if you execute this, it will show you the tables. So here you can see that there are three tables. There's a table for dataset level information on metadata. There's a table for sample level metadata, and there is a different table for single cell metadata, single cell sample metadata. And there is a separate table for single cell because single cell is stored in a different file format, and which is H5AD, whereas the other data types are stored in GCT. So there is two different tables for sample level metadata that is present in this omics atlas. Next, let's just see what columns are available in this table that contains dataset level metadata. So for that, you need to run a describe query in SQL. So it goes like this: describe omics atlas name and then the table name, which is datasets. Provide the query to the function and run it again. So on running it, you can see that here there is an output where there is column name and column type here. So the columns that are available are organism, total number of cells, total number of samples, gene modified, description. This is more of a paragraph column. So this is raw. And this is the raw text data that is taken and stored. And then there is publication, there are other columns for disease, drug, etc. And then there are data types as well. So here you can see this column type is array string. What that means is in this column, the data can be stored or the values can be stored in form of a list. So there need not be one value per row for this column. It can have multiple columns per row. And then there is integer data type, there is string data type. In these columns, there'll be one value per row. you cannot have multiple values per row for these columns. And there is float data type as well, but it's not there in this particular schema. So similarly, you have tables for samples and for single cell samples. You can run these two cells and view what columns are available. And next, we can query the data sets itself. So here again, we are querying data sets related to Alzheimer. So here we can provide a SQL query where we say select these few columns, which is data set ID, curated disease, data type, data set source from mocktherapeutics.datasets, which is a dataset level metadata, where it contains curated disease as Alzheimer's. Now, there is a function contains used here for getting Alzheimer's disease dataset. It's because this column curated disease is of array type. So it has list of values. So in order to get datasets for which this list of values has Alzheimer's, you have to use this contains function. And next there is recommend function, which takes in a keyword. So here you can type Alzheimer, or you can also try typing brain. And there are these two parameters, which is related, and then the column name that we are querying. And what this does is it just fetches all relevant terms that are needed for the search, and it conducts a search and gives out the data sets. So for example, if you're searching data sets for breast neoplasms, then that can be represented in different types. So there can be breast neoplasms, there can be triple negative breast neoplasms, there can be cancer of the breast carcinoma. So all of the terms are fetched by this recommend function, and those are used in the query automatically, and you can fetch the results. So you can run this, and it will give you 37 rows, which is so here we can see that there are these many data sets for Alzheimer's disease, and they are of transcriptomics and single cell data type, and they are from the source GAO. So moving on to the next query, let's say we want to know what is the number of data sets for each data type in Alzheimer. So here we can select the few columns that we want. Additionally, you can also provide count bracket star, which counts the number of data sets. Here I have selected data type and disease name. The names of the columns are different. It's because I am splitting the list into individual values, and in the query later on where I've given cross join unnest. So here what this does is it splits the value in the list or in the columns that have the list data type, and the data that it displays out will have only one value per row for these columns. So this has been done for both the disease and data type column, and the columns in which there is individual values per row, those column names have been provided over here. And then again, we are saying give me data sets for the disease Alzheimer's, and group them by data type and disease name, and then count the number of unique combinations and order them in descending order. So once you execute this execute this cell or run the cell, this will be the output you get. So we can see that for the disease name Alzheimer's disease, we have 30 transcriptomics data set. And again, for single cell, we have 7 transcriptomics data set. 7 single cell data sets. I see a question in the chat, so I'm just going to go there and see what it is. Can you please repeat the steps after creating workspace again? These steps are there in the workspace Amity Uni workshop. So there's a file called Amity README. You can just click on it and preview the file by clicking on the eye-shaped option over here, and it will have all the steps to open up the notebooks. And yeah, so moving on to the next query and the last query. So suppose we want to find case control Alzheimer disease data sets. Case control would mean that we also have normal samples in the experiment. So the experiment compares the condition normal and the diseased condition and finds out variations in the diseased conditions. So for this, we will be querying the sample level metadata. So select a few columns from the sample level metadata, unnest it, which is make the list as one value per row, where the disease should either be Alzheimer's disease or it should be normal. So this is the term we use to tag samples that are healthy or normal or that are used as control. We only use the term normal to tag such samples. And then additionally, we also give a condition saying that the dataset ID should be among the datasets for which the disease is Alzheimer's. So once we run this, we get this output where we have multiple samples, their corresponding disease, and I mean multiple datasets, multiple sample IDs, which is the sample names, and their corresponding disease tag. So once you have this, we need to find which datasets are case control. So to find the case control dataset, we cross-tab these results. So what crosstab does is it just displays the number of samples that are there for Alzheimer's disease and the number of samples that are there for normal. And here it also displays the dataset ID that they come from. And the next condition is that among these two columns, which is Alzheimer's disease and normal, neither should be 0. So if either Alzheimer's disease is 0 or normal is 0, then those dataset IDs are filtered out because they either don't contain a normal at all, or in the dataset, all of the samples are tagged as normal. So once you execute this cell, this is the result that is displayed. And we can see that there are two datasets that are case control. And we can take these datasets forward in the analysis that we want to perform. So this is a very gentle introduction to how you can narrow down and find data sets that you want for your analysis on poly. And if there are any doubts, if there are any questions that you want to ask, please feel free to ask me. So any questions, any doubts?
I don't think there are any questions anymore.
Okay, so I hope this session has been helpful, and I hope the session has shown you how Poly makes it easy to find data sets.
We have one question. How can we have access to Poly? Like, I think without the Amity ID, how can the students use it independently?
Well, your access to Poly requires a subscription to Poly. So you'll have to have that subscription in order to have a login email, and then you can use Poly after getting that subscription. For that, you'll have to reach out to us. And
I think that's all. So thank you so much for such a wonderful session. It was really insightful. Okay, one of the students has said that she cannot find the README file.
It's there in the Amity _ workspace. It's there in this workspace, which is Amity Uni -workshop. And there's a file named Amity _readme.csv.
Also, does this, the link that you've created, the email that you've created with the Amity University, does it have some time duration or it's always valid? The files will always be there?
It definitely has a time duration. So I don't think this ID can be used after these sessions are done. So if any of the notebooks in the analysis sessions that are being held after this are useful to you, you can download the notebooks and you can refer that analysis. Although you won't be able to access Poly data, you can use the same analysis on the data that you might find externally.
So again, thank you so much for joining us today. Thank you for such an insightful session.
So for anyone who might have further questions,
there is another question in the chat box. How to copy this file to my workspace?
It's there in the README file. Please read through it and follow the steps. So yeah, I hope this session has been informative. And if there is anything more you want to know about what we do, anything more you want to know about Poly, you can contact me via mail. I'm dropping it in the chat. It was nice to have this session with you all, and I hope the next few sessions will go well as well. So thank you very much.
Thank you so much. So, ma'am, I think we do have Mr. Harsh with us. So should we move on to the next session?
Hello. Yeah, sure. We can move on.
Okay, so you're ready?
Yeah.
Okay, so I'll just give a brief introduction to the participants. So our next session is on single cell RNA-seq data on Poly by Mr. Harsh Malviya, Elucidata, New Delhi. Mr. Harsh Malviya is a data analyst in the solutions team of Elucidata, where he works on generating technical demos of the platform Poly to showcase to the potential clients and supporting the team in solving other bioinformatics related challenges. Academically, he has a master's degree in biotechnology and a postgraduate diploma in big data and biology. So I would now request you to please start the session.
Yeah. Hello, everyone. Thank you so much for inviting me and for that introduction. So, hope I'm loud and clear. So I'll share my screen now. Okay, my screen is visible, right?
Yes, it's visible.
Yeah. So hello, good evening, everyone. As you have been introduced to me earlier, we can start with the session. So first, I would like to get an idea about the knowledge base that we have as a group here about any sequencing technique. So can people reply to me in the chat and let me know if they have any idea or even a little bit experience with any analysis of bulk RNA sequencing or single cell RNA sequencing?
The participants can write yes if they do have experience.
Yeah, so okay. I would really like if people would reply at regular interval in the chat, because this is going to be a little bit, this session is going to be a little bit more technical, and I want to keep everyone on the same page so we can have a good session. Okay, Niharika is analyzing bulk RNA-seq data. So yeah, okay, we can move on, I guess. So no worries, even if you are new to single cell or any RNA-seq, I will be trying to cover the top concept of single cell RNA-seq data as fundamentally as possible. Yeah, so first we'll look at a general introduction to single cell RNA-seq data. So when we when we talk about RNA sequencing conventionally, we are referring to bulk RNA sequencing. So as it might be clear from this analogy on the slide, so bulk RNA sequencing is like a smoothie in which we are combining the mRNA of all the constituent cells from the tissue that we have isolated, and we are looking at the average and we are looking at the overall transcriptomic profile of the sample or the tissue. While in the case of single cell RNA-seq data, we are isolating each individual single cell and then studying the transcriptomic profile of each individual single cell, which gives us a higher level of granularity, and we can ask more specific and granular biological questions. So what kind of questions can we answer using scRNA-seq data? So as we are isolating each individual single cell and looking at the transcriptomic profile, we are able to identify different cell types which may constitute a tissue or a sample. We are also able to look at the rarer type of cells, which may be less in number and overshadowed by other types of cells which may be more commonly present in that particular tissue, but oftentimes it is the rarer cell types which are more biologically significant for a question for research purposes. Also, in the case of single cell sRNA-seq data is oftenly used in studying immune cells, because immune cells, as you might know, undergo a clonal evolution on stimulus, and using sRNA-seq data, we are able to follow this clonal evolution and then able to compare the progenitor cell with a more differentiated version of the same cell. So this is an application which is often used in studying autoimmune disease, or the role of immune cells in autoimmune diseases. Single cell sRNA-seq data also allows us to identify biological markers or transcriptomic markers, which are often missed by just looking at the expression of marker genes in case of bulk RNA-seq, or even while looking at the cell morphology. And using this, and by defining these transcriptomic signatures for individual cell types, we can then group different cells in a disease condition and identify a new subpopulation of cells which may be playing a role in the particular disease. So this is a general workflow of sRNA-seq analysis. Starting with a solid tissue, we first dissociate the individual cells, and then we carry out single cell isolation. So this single cell isolation can be carried out using two main types of techniques. It may be plate-based isolation methods or droplet-based isolation methods, which I'll be talking about more in the next slide. So after we have isolated each single cell, we carry on with the normal bulk RNA sequencing library preparation steps, where we take the mRNA, convert it to cDNA, then amplify the RNA, and then form a library. Then we load it into a sequencer, which is oftentimes the same sequencer which is used for bulk RNA sequencing only. But in this case, in the case of single sRNA-seq, at the step of single cell isolation, we make sure to incorporate a specific nucleotide sequence, which corresponds to a particular cell, which is called a barcode. And this barcode would be attached to each mRNA molecule of the corresponding cell from where the mRNA came. And using this barcodes, we are then able to separate out each mRNA molecule and group them based on their source cell. And by that way, we are able to generate single cell expression profiles for each individual cell. And using this expression profile, as I mentioned earlier, we are able to identify different cell types. So these are the main techniques which are used for single cell isolation in sRNA-seq analysis preparation. So as I mentioned earlier, this is we can pipette out in each individual cell and culture them in a 96 well plate, where each well contains a single cell. We can also use FAS or fluorescent assisted cell sorting to separate out the same type of cells into different wells, and then again using a pipette or micro injection separate out each individual cell. While more modern methods and more common methods which are used now, such as 10x genomics, if you may have heard the name, use a microfluidic or droplet-based method, wherein cells from a suspension are flow into a channel, and then they're captured inside a droplet, an oil droplet, and this oil droplet consists of a bead which has the cell barcode and a primer which attaches to the mRNA of that particular cell. So after cell lysis, each mRNA molecule of that particular cell has a barcode and a unique molecular identifier attached to it, which then allows us to later identify the source of that particular mRNA molecule and form expression profile for that particular single cell. So I hope everyone is following. I can go slower if there is a need. Okay, and let's move forward. So this is an overview of the actual analysis which goes into single cell data. So after the sequencing is done and the raw data processing is done, which includes removing cells, which includes removing libraries which may not have found properly and other such technical errors. We are presented with a count matrix, which, as Anaga described earlier, has a dimension of cells by gene. So, or it can be genes by cells by gene or gene by cells. Then we perform a few quality control steps which are specific to single cell RNA analysis, which I'll be talking about more and showcasing in the hands-on part. After that, we perform clustering, and then the clustering part mainly allows us to identify the different cell types. If we don't have any knowledge of the cell types that we have isolated, we can identify the different cell types using clustering methods. And then after we have identified our different cell types and formed clusters, we can do more downstream analysis and ask biological questions by comparing the clusters. We can do differential expression analysis between two different clusters or between clusters belonging to different conditions, or we can do trajectory inference, as I mentioned earlier. In the case of immune cells, or stem cells, we can compare stem cells with transcriptomic profiles of stem cells with progenitor cells, and as such. We'll also look at this analysis in the hands-on part. So coming to how single cell data is stored on Poly. As Anaga mentioned in detail, how our process of curation and storing data. This slide is particular to single cell data. So we have a list of sources from which we get our data sets. We can get it from these public repositories, or even a file storage system which a client might have. And after getting the data sets from these sources, our curation models work on the data sets and standardize the metadata of each of the single cell data sets. So all of our single cell data sets have follow a standard nomenclature and are processed in the same way, making them easily comparable. After the standardization is done, we push the data to the enterprise atlas, which is the same thing as what mock therapeutics, which we are going to use today. And after the data set is on an enterprise atlas or a omics atlas, we can then access the data set using the Jupyter Notebook environment, which we'll be showing today, and perform downstream analysis on the data set. And also, if there is a requirement, we can generate an API for used by clients on their own platforms. So as I said, we don't just copy paste the single cell data set from source to our atlas, omics atlas. We perform a standardized set of pre-processing steps on each data set as to ensure quality and comparability, consistency between different data sets. So we perform a few QC steps which are filtering. So we remove cells with too many mitochondrial genes or high counts, or genes which are found in less than 3 cells. So we'll be talking about this more in the hands-on session again. And we also perform certain basic analysis and store it in the data sets like PCA, like generating PCA and UMAP and TSNE. Another thing that we curate is cell type annotation. So many publications do not have a cell type annotation when they submit the data. So in that case, we run our own analysis and identify the cell types. And in the case there is a cell type data annotation data available in the data set at source, we then validate if that cell type annotation is correct or not using clustering techniques in our own analysis. And in the case the single cell data which at source was already in the form of process count, we can skip the QC step and just go with computing the PCA, the UMAP, and the cell type annotation. So yeah, are there any questions in the chat? Or?
I don't think there are any questions in the chat box. I think you can move on.
Okay, so I'll go ahead then. So let's start with the hands-on part now. So I hope everyone will respond in the chat now on, so no one is left behind. So this is the homepage that you all might be seeing when you log into your Poly accounts. So everyone was able to do this step, right? Or is anyone else still stuck at logging in part?
I would encourage the participants to please respond. Even if you have already access, just type a yes so that we could know.
Yeah, is it, can...
Yes, now we can see.
Yeah.
I don't think anyone has any doubts. So I think everyone has access.
Okay, we'll go ahead. So Anaga has already introduced you to the basic elements of the UI. So we can again look at how we can query single cell data using GUI first. So we go to the Omics Atlas tab, and then we go to the mock therapeutics Omics Atlas. And you can see here that we have 23 single cell data sets. So let's assume we are trying to look at data sets for a particular disease which is relevant to our research. So I want to look at the single cell data sets for single cell data sets which are based on neurodegenerative diseases. So first I'll select the data type here as single cell. Then I can look at neurodegenerative diseases. And then I also want to filter it down to data sets where the tissue is prefrontal cortex. So this is the data set that I get. So if I want to look at the sample level information for this particular data set, so I would like to point out that all these tags that we see here has been curated by our machine learning curation model, which has tagged this, which has looked at the descriptive fields of this data set at source, which is on Geo, and tagged that this data set is associated with neurodegenerative diseases, alcoholism, it is the organism is Homo sapiens, and there are different cell types such as endothelial cell and excitatory neuron. So we can go ahead and look at the sample level metadata. So here I get more detailed information about the data set. So this study is based on profiling single cells from the prefrontal cortex of humans suffering with alcoholism, and also comparing them with control subjects. So there are 8 samples, and from these 8 samples, we have 39,000 cells. You can see here that 26% of the samples belong to the alcoholism cohort, while 73% is the normal cohort, and all the samples are from the prefrontal cortex. Okay. So if you want more information about this data set, you can click this link below the source, and it will take you to the PubMed page of the publication from which this data set comes. So everyone still with me? I hope. Okay, so now we can go on to the notebook part. Yeah. So there was some confusion about how to copy files from one workspace to another. So I can go over that part again. So the notebook required for this session is in this folder called Single Cell RNA-seq Workshop. So we go inside this notebook and inside this folder, and we can see this notebook. So I would like everyone to copy this notebook to their own workspace. I can see very few workspaces compared to the participants. Is there any issue in making workspaces or logging in? I also demonstrate it again. I'll make a new workspace. We go to the source workspace. We go to this folder. We click on the notebook. And on the right hand side, you will be able to see the copy option. Here in this tab, you select your own workspace. I can select my workspace and then copy files. It says successfully copied. So now I can launch the notebook from my workspace. So since we are going to, analyze single cell data, I would suggest everyone to use this polyglot Docker. So the advantage of using a polyglot Docker is that using this Docker, we are able to run multiple, we are able to code in multiple languages at the same place. So people familiar with bioinformatics analysis may know that some tools require R, while some tools may require Python and some tools may require a Bash environment. So using this polyglot environment, we are able to code in all these 3 languages in the same Jupyter notebook. And for the machine type, I would like you to select poly extra large because the analysis can be a little bit resource intensive. So the CPU and RAM information is undergoing some changes and is incorrectly displayed right now. It is 16 GB RAM and 8 vCPU. So just select poly extra large and click on launch. So can I get that done from people who are able to successfully launch the notebook? A few more replies and then we can continue. Select the poly X large machine, and this one. Okay, I guess we can move on now. So yeah, again, this environment is the same as what Anaga showed you, and this the Jupyter notebook environment is versatile enough to perform multiple type of analysis on it. So again, we in this tab, we are installing certain packages that will require for carrying out our downstream analysis. the main package is ScanPy. So ScanPy is a widely used Python-based single scRNA-seq downstream analysis package. You may have heard about other R-based packages like Surat or Scran. Please make sure you run this tab. So on this side, you can see that in this drop-down menu, we have this option of these 3 languages. So as I mentioned earlier, each of these cells can be configured to run either of the 3 languages, that is bash commands or Python or R. And polyglot will just detect what is the language you are coding in. So when we are going to be using Python only for this workshop, but the same concepts and same steps can be translated if you are going to run your analysis in R using Surat, which is also a widely used tool for scRNA-seq analysis. And ScanPy is based mostly on Surat but converted into Python. So in this cell, we are importing all the modules required. And then these are some visualization settings that we are setting up. So SC here means ScanPy, since we have imported the ScanPy module or package as SC. Every time we write SC, it will understand that it refers to a ScanPy function. So here we are setting the ScanPy settings verbosity to 2. So it will display various information about what the code is doing at the moment. And we can disable it if we do not want to see any outputs from the function. At this here we are using the authentication, getting the authentication for using the polyomics atlas. And this is an overview of the curation that we do for each of our data types, each of our data set regardless of the data type. So we curate the disease, tissue, cell line, drug, organism. So the nomenclature is uniform across all the data sets, which makes querying across different data types and across different sources very easy. We also do curation at sample level for each of the sample which in the data set. So this cell again shows what are the tables which are associated with this mock therapeutics omics atlas. So the dataset level metadata is stored in this table called .dataset, and sample level is stored in .samples, while single cell sample metadata is stored in this different table which has a .samples underscore single cell. So we can use PolyPython to query the same dataset that we queried using the GUI by writing a small SQL code. So here I am selecting the columns which I want from the table, and the table is mock therapeutics.dataset. So I'm looking at dataset level metadata. I want dataset where the data type is single cell, and the curated disease is neurodegenerative disease, and the organism is Homo sapien. And I want the results to be sorted by total number of cells. So it gave me 2 results. We can see. So this is the data set that we saw on the GUI. Now we can look at the metadata associated with that particular data set again. So we have already discussed this data set while using GUI. So you can go ahead. So now I want to look at what type of metadata is associated with each sample of this data set. So I will query the sample level metadata of this data set from .samples underscore single cell, and it gives me 39,000 rows. As we saw that we have 39,000 cells associated with the data set. So here we can see the different metadata associated with each of the cell. So you can see that there is alcohol use disorder, there is age, there is sex, and we have the curated cell types also for this data set. So in this data set, the cell type annotation was present in the source metadata, but again we have, as I mentioned in the presentation, we again validate whether the cell type annotation was correct for that particular cluster, was correct by performing clustering, and then from that we get the curated cell type. And here you can see the associated barcode for each of the sample and the title. So now we can go ahead to the actual analysis part. So Anaga has already given a brief overview of the H5D format. So after our connectors or our models are done curating the data sets, we save each single cell data set in an H5D format. So an H5D format is a format same as a CSV file or something, but it is more complicated to support storing various metadata along with the actual count data of the data set. So we have 4 main objects associated with H5D file. Here we have the main object, which is .x, which contains the actual count matrix of how many counts of particular gene in a particular cell. Then we have the .var, which contains information regarding the metadata regarding the variables or features, which is genes in our case. Then we have the .obs object, which contains the metadata regarding each observation or cell in our case. And then we have the unstructured object. So the unstructured object is mainly used to store extra information, such as if we have conducted some certain analysis and we want to store it with the data set. So in order to utilize it at a later point, we can store it in the unstructured object. And you can see here that .var and .obs are pandas data frame, while .x is a numpy array or a sparse matrix. So generally, depending on the source of the single cell data set, we can we store the count matrix in a sparse matrix. So since for single cell data set, most of the count for genes will be 0. There will be a lot of 0 in the matrix. So there will be a lot of 0, 0, zeros, and there will be then a particular gene in one of the 35,000 cells will have a count of 2 or 3, but there will be a lot of zeros. So in order to save memory, because these 0 counts, because saving these zeros is not of importance to us or not useful for our analysis, this sparse matrix just stores the positional location where the count is not 0. And using that, we are able to efficiently store such a large matrix. So imagine a CSV file which is 39,000 by 34,000. So that would make a huge CSV file. But using this sparse matrix, we are able to reduce the size and use it more efficiently for our analysis. Okay. So now at this step, we are downloading this H5D file for this particular data set from the omics atlas. and loading it into a Jupyter environment so we can go ahead with the analysis. So I want everyone to run this cell. And again, let me know in the chat when you are done. So please let me know if people are able to download the dataset. I'll just wait for a couple of more replies. So if you're wondering that what will we do, good. So if you're wondering that how at source we don't get H5 files and what is the user on H5, you can use a package called NDATA which allows you to load files attach the various.
I think there is a little technical issue with your audio. It's slipping. Your voice is breaking.
Oh, is it better now?
Yes, it's better now.
Yeah, so using the end data package, you can make the file from the various you can get from GEO, which may be if the data set was created in 10x cell ranges, or it can be if
your voice is still breaking.
CSV. Let me change my internet.
Yeah, it's still breaking.
Is it better now? And
a little better, but I think it's still a little. Now your video is also not moving.
Hello, hello.
We can hear you just a little in between.
Oh, strange. So is the screen moving fine? So if I scroll down, so it's only and the video is also fine, or is it also lagging?
It's a little laggy.
It's a little laggy. Okay. Maybe the internet is raining over here. So is it bearable? Like if I talk slowly,
it's better
can
now.
we go ahead?
It's better. Yeah, we can go ahead. Thank you.
Yeah. Thank you so much. So I hope like a few people are done downloading. So yeah, in ScanPy we have this read H5 function which will read the file. So if we run this, so it's better if I don't run any commands right now because of my laggy internet, but yeah, I can still explain them. So so people who are following along would have gotten this output here as the file got loaded into a variable called AData. So AData stands for end data object, which is the H5 file type. So here you can see we get a summary of the dimension of the actual file. So as I mentioned earlier, we have a matrix of 39,000 by 34,000. So we have 39,000 cells and expression information for expression data for 34,000 corresponding genes. In the OBS object, we have various metadata fields and various metadata fields which are related to the observations or the cells. So here we also have our the curated fields that our ML model curates. So we have the curated drug, cell type, and curated marker present or absent, the disease and the tissue. Then in the var object, we have metadata associated with the variables or genes. So we have gene IDs, we have feature types, and other associated metadata. So since data sets on poly are already pre-processed, we have a few more metadata associated with in this end object, if compared to what you might find if you had to generate your own H5D files. But all of this fields are a part of and you will be adding them one by one. So as you can see here, we also have information for the PCA, the TSNI, and the UMAP. Okay. So we can look at each of the object again. So we can look at the OBS object, and so here using Plotly, I have plotted a sunburst diagram of describing the cohort in this data set, which makes it easier for us to understand what kind of analysis or what kind of questions we can ask from this data set. So here we can see that we have two main cohorts, which is normal subjects and patients from an alcoholism cohort. We can see the counts of each cohort here, and associated with each cohort, we have the various cell types. As you hover over them, you can look at the cell types and the count. Since we also already have this cohort information, we can also look at the proportion of cells in each of the cohort. So we can look at the proportion of GABAergic neurons in our normal cohort versus our alcoholism cohort, and so on. So this type of plots help us understand whether this particular data set is relevant to the research goal we have. Now we can start by looking at quality control in pre-processing steps. So there are three main QC metrics in case of HCRNA-seq data, which are gene counts, UMI counts, and percent mito. So as you can see, gene counts is the number of unique genes which is detected in each cell, and UMI count is the number of total molecules detected within a cell, while percent mito is a percentage of mitochondrial genes. So what the problem with single cell data is that sometimes we may get cells which have died, or sometimes the barcode may go into an empty droplet while performing the single cell isolation step, and there may be no genes associated with particular barcode, but it will show up as a cell. Or other case that can happen is that two cells can enter inside a droplet, and we get a very high count for that particular barcode. So we need to QC, so we need to remove those cells using these QC metrics. So as I mentioned earlier, our pipeline automatically performs the QC, so we don't have to do it for this data set. But the main QC steps that we do is that we try to filter cells which do not have a very high gene counts, which may be representative of doublets present in a single, which may be representative of doublets. We also limit cells with less than 25,000 UMI counts, again to prevent doublet detection. And again, percentage of mitochondrial genes. So as a cell undergoes lysis, the proportion of mitochondrial gene increases inside the cell. So cells associated with high percentage of mitochondrial genes are probably lyse cells or dead cells, which are of no use to us in downstream. So we want to keep the percentage of mitochondrial genes less than 5%. So you can see we have maximum of 2% mitochondrial percentage. Then we can also look at the highest expression genes with the highest expression. So already in this case, I have done the pre-processing. But generally what we would see is that in this list, there would be MALAT1, which is a gene which is highly expressed inside, which is ubiquitously expressed in cells and associated with cell lysis and other mitochondrial genes. So what we want to do is that we want to filter out those cells before we continue with our analysis. So here we are storing a list of all the genes that we want to remove in variables. So we're looking at gene names which start with MT because those represent mitochondrial gene and MALAT1. And we also want to remove ribosomal genes. And then we slice our data in order to remove all those genes which may indicate that the cells are undergoing lysis or are of low quality. So these are the commands for all the pre-processing steps that I showcased in the presentation and which are done automatically. So this is for reference of the participants. We don't need to run these commands for this workshop, but if you are trying to perform your own analysis, it may be helpful for you. Okay. I hope everyone is following along. I think I need to rush because we lost a bit of time. So I'll just go over all the plots quickly and we can take questions at the end, or you can just leave them in the chat. So in case of single cell data, we want to focus on we want to reduce the number of genes that we want to reduce the number of genes that we are taking into account for our downstream analysis, because there are a lot, in order to reduce our dimensions. So what the most common method of reducing dimensions in case of single cell analysis is to look at highly variable genes. So highly variable genes are those genes which are responsible for cell-to-cell variation because of their biological effect and not because of their increased expression or increased size. So highly variable genes are detected by modeling the mean variation relationship of a gene across all cell types, and then by selecting genes which show a variation above the predicted threshold, which is decided by the model. And that's how we get our highly variable genes. So next method of reducing dimension is again the aim of dimensionality reduction is to reduce the amount of data that we look at to find significant information, or in a short way to reduce the amount of noise. So another most common dimensionality reduction method is principal component analysis. So PCA is used across all types of machine learning and other analysis, also bulk RNA-seq or any other form of data analysis where you need to reduce the number of dimensions. Okay. Again, the goal of PCA is to reduce looking at 34,000 genes and instead only focus on principal components which account for the highest variability in the data set, because we are only interested in what is causing the PCA. So we are arranging each of the cells according to their variation along these two principal components, which account for the highest amount of variation in the data set. And we can just focus on this component, on analyzing genes that contribute to these components, rather than looking at all the 34,000 genes. So here you can see the cell types. Since we already know the cell types, I have colored each individual cell according to the cell type they belong to. But in case of so PCA is useful, more useful for data which can be explained linearly, or which can be counted by a linear function. But single cell scRNA-seq data is more often explained by non-linear functions rather than linear functions. And so in the case of scRNA-seq, we use other dimensionality reduction methods such as t-SNE and UMAP, which are non-linear and which can capture non-linear data and allows us to identify the spatial arrangement of the cells in a better way than PCA. So as you can see here, the t-SNE plot looks way more organized than the PCA plot, and the clusters of each different cell types are more clearly defined. So similar to t-SNE is UMAP. The difference between t-SNE and UMAP is that t-SNE tries to conserve the local similarity between the local areas more, and tries not to look at the global similarity. So it does not care about the similarity of this cluster with this cluster. It just focuses that in this local vicinity, the similarity is high. But so t-SNE optimizes for local structures. However, UMAP is an advanced method which tries to preserve both the local and the global structure of the data. So another thing I like to note is that t-SNE and UMAP are both stochastic methods and based on neighborhood graphs. So you might feel like I'm throwing around these terms like neighborhood graphs and stochastic methods, but the truth is a lot of high-level statistics goes into performing scRNA-seq analysis, and I wish we had more time to discuss all that, but I can just give you a brief overview of the terminologies today. So by stochastic method, I mean that the formation of t-SNE and UMAP both start at a random point from the data set. So every time you run the t-SNE command, it might give you a different graph because it started looking for neighbors from a different point. So in order to do that, we set a random state here. So I have given an argument random state equals to 4. So it knows that every time I run this command, it has to start from the same point and give me the same output. Similarly for UMAP. Now we go ahead with clustering. So in the case of this data set, we already know what is the cell type of each cluster that we are seeing. But suppose there is a data set where we don't know, or we have no cell type annotation. So in that case, we can perform unbiased. We can use this unbiased method of clustering to identify the different cell types. So here ScanPy enables us to use a graph-based clustering method called multiple graph-based clustering methods such as Leuven and Leiden algorithm. So Leuven algorithm is widely used in publications till now, but the latest version of ScanPy has implemented a Leiden clustering method, which is an improved version of the Leuven algorithm. So yeah, we can go ahead with using the Leiden algorithm to perform the clustering. Here the resolution argument resolution argument represents the amount of granularity or fineness or coarseness that we want in our clustering. So what it simply means is that it corresponds to the number of clusters that we would like it to make. So if I were to increase this value here, it would form way more than 8 clusters, and cluster even 2 cells, 3 cells together. So this resolution value is actually something we need to identify by running while doing the analysis. So we can start with any value. So generally, it is suggested to start with a value of 0.1 and perform the clustering. Then if we have a ground truth cell type label available, such as in this case that we already know what cell type each cluster belongs to, we can use the rand index to compare if our unbiased clustering is similar to our ground truth or not. So here sklearn is scikit-learn, another Python package which is widely used in machine learning, and it has a function for calculating the adjusted rand score. So here we give it that labels true, which is the ground truth that we know that this particular cluster 8 is oligodendrocyte or a microglial cell. And then using statistics, it is able to calculate an index which represents whether our unbiased clustering is similar to our ground truth or not. So we want this index to be as close to 1 as possible. So as I said earlier, you can start with a resolution of 0.1, then calculate your rand index and look at the value. And then you can try slowly increasing or decreasing this resolution parameter to get the maximum possible rand index. So I was able to get 0.81 using the value of 0. Now that we have our clusters and our cell annotation, we can go ahead with our downstream analysis and asking actual biological questions in the form of differential expression. So ScanPy allows us to carry out differential expression using various statistical tests such as t-test using this command, where we are telling it to perform differential expression or perform the differential expression analysis of each individual cluster against the rest of the clusters. And then using this plot rank genes group, we are able to see the plot. So this is the differential expression analysis for cluster 0 compared to the rest of the clusters. And we can see here that this gene CAM1, MAP1B are highly expressed, highly differentially expressed. Similarly, we can see the results for each of the clusters. And this information that this differential expression results that we got is stored in this key variable that you can define. So I have defined it as t-test. And if you see here in the unstructured object, as I mentioned earlier, we have stored the t-test results here. And using the unstructured object, we can again use this t-test results. So here we can get the top 5 differentially expressed. False. Yeah. So I think I'll take questions at the end, just in the favor of time. Yeah. So yeah.
There is one question.
And then we also have other statistical methods like t-test. Yeah. So we'll just finish the notebook once and then we can take questions. So yeah, similarly, we can carry out differential expression using multiple methods. So this will answer Jitendra's question also, that instead of relying on the results of just one test, one type of statistical test, we can carry out differential expression using multiple tests. So t-test and t-test overestimated variance are two parametric tests, while Wilcoxon.
I think you're not audible. I'm not sure about the other participants, but I think you're not audible.
Hello, ma'am. Ma'am, Sudipti, ma'am.
Yes.
Ma'am, I think there is some network issue from Harsh, yes, Harsh sir, and
I think Harsh, so you can, I think, turn off your video if the bandwidth isn't that strong enough.
Sir, if it is possible, you can rejoin. If it is possible from your end.
He'll have to be made the co-host again. Just.
Ma'am, can you type in the chat box to sir? I think
I have.
Yes.
I have typed it in the chat box.
Yeah, so after we are carrying out differential expression analysis with multiple tests and taking the results which are common to all the tests, we can move ahead with visualizing the differential expression. So here I have just gone ahead with using the results of the Wilcoxon test, which I think are a fair representation of what was published in the publication also. And we can look at various visualizations indicating the differential expression of the genes. So here we are seeing the top 5 differentially expressed gene for each of the clusters and their expression in other clusters. So here is the top 5 differentially expressed gene in cluster 7, and you're looking at their expression in the remaining clusters. We can also plot a scatter plot and visualize the same thing. Again, how the expression of the differentially expressed gene in a particular cluster varies in the remaining clusters. So here we can see that this particular gene LHPLR3 is totally totally unexpressed in cluster 11, while the remaining clusters show different varying level of expression in a So here we carried out the differential expression analysis for each of the clusters and comparing them with the rest of the clusters. We can also, since we have the cell type annotation already, we can perform the same analysis for each of the cell type also. So these plots show the differentially expressed gene in a particular cell type compared to the rest of the cell types. So for GABAergic neurons, we can see that CNT and AP2 and these genes are differentially expressed. Similarly, for all the other cell types, we can see that. We can plot a scatter plot or dot plot, and see how the expression varies across different for differentially expressed gene varies across different cell types. But more importantly, we are generally more interested in studying differential expression across different conditions like control versus disease. So in this case, we have 2 cohorts, normal and alcoholism. So here we are fixing a particular cell type. So we are taking cluster 0. So we can see here that cluster 0 corresponds to excitatory neurons or oligodendrocytes. So this is the cluster the publication was also interested in studying the most. So we can so we have sliced our data such that now we only have data for this. Was my screen not visible?
It is visible.
Okay, the Zoom is giving me a prompt now. So yeah. So we have sliced the data such that we have counts for only that cluster 0, and we are going to group the samples based on the curated disease cohort. And again, we can run the differential expression analysis using any statistical test that we would like. So I have again used Wilcoxon test here. And we can see here that for the oligodendrocytes and excitatory neurons, we have these genes which are differentially expressed in the alcoholism samples from the alcoholism cohort and these genes from the normal cohort. So we can then go ahead and plot this results as a split Violin plot. Yeah. So here we can see that in the case of, so the blue side Violin plot is the expression of the genes in the alcoholism cohort, cells from the alcoholism cohort, while the orange side is from the normal cohort. So we can see that there is increased expression of the differentially expressed gene in the alcoholism cohort. We can also look at the expression. We can also look at the same thing for cell genes differentially expressed in the normal cohort. So yeah. Another thing that we can do is we can plot the expression. We can look for the in this case we looked at differentially expressed gene in only one cell type and compared the expression between the alcoholism cohort and the normal cohort. But since now we have our list of differentially expressed gene, we can look for the expression of these genes across the rest of the cell types also. So we can plot a cat plot using SNS Seaborn library like this. So as you can see here, the cell types are mentioned below, and we can compare the expression of the differentially expressed gene BCYRN1 in alcoholism cohort and normal cohort. Yeah, and from such visualizations, then you can pinpoint the particular gene or particular genes you want to go ahead with, and then also look at pathway analysis using GACA and other methods. But for the sake of our workshop, this analysis demonstrates the most of the ScanPy's functionality. Yeah, so that is for this part. I hope in spite of the technical difficulties, the participants were able to understand some of the analysis and learn something new. We can take questions now if we have time.
We do have like 5 minutes if the participants want to ask their questions through the chat box.
I'm not able to see my chat box, so can anyone read the questions out to me?
So there was one question: which method do you follow to control false positive signals in differential gene expression? Did that get answered yet, or like?
So as I mentioned earlier, that for controlling for false positive, because all of these differential expression tests are probabilistic methods. So the only way to control for false positive is to perform differential expression analysis using multiple types of statistical parameters and test, and then just using the genes which are differentially expressed among in multiple tests, like genes which are showing differential expression in t-test also and Wilcoxon test also, and just going forward with those genes as differentially expressed.
The doubt is that if you perform multiple tests, chances of getting false positive signals is even higher.
Yeah, but we are taking a union of these tests. So we are controlling for the fall. So the different, and we are trying to incorporate different types of statistical tests. So as I said, the t-test and the t-test overestimated variance test are parametric test, while Wilcoxon test is a non-parametric test. So we are considering different statistical assumptions also. So t-test and t-test assumes a normal distribution, while the Wilcoxon test does not assume a normal distribution of the data set. So even then, under different assumptions, the a gene is showing as differentially expressed, we can be more certain that it is not a false positive. And again, after getting the test, we can look at the plots and visualization methods and again filter out cells that you see that the ground truth is there, that the expression is more in our cohort compared to the reference cohort.
Okay, so I don't think there are any other doubts.
Yeah. So I would suggest the participants to, if they would like to download and keep a copy of this notebook for their future reference, because they might not get access to their Poly accounts, but they can still use this notebook in their local environment. And this tutorial has been based on mostly on these 3 tutorials which are present on the internet from reputable institutes like the Chen Zuckerberg Institute and NBI Sweden, and also the ScanFi's official tutorial. So yeah, they can use it as a reference. So in order to download and keep the notebook, you can go to the file and download as not a notebook and store it in your local environment.
Okay, so thank you so much, sir, for such an insightful talk. I'm sure the participants really benefited from all these steps that you showed in such detail. It was great having you.
Yeah, thank you
We
so
really
much.
hope to see you again.
Yeah, sorry for the technical disturbances.
No, that's okay. Sometimes it's really not in our hands. Thank you again for joining us.
Yeah, thank you, everyone.
So we'll move on to our next session, but before that, just a quick couple of announcements. So first, I would be soon sharing 2 kinds of links. One will be the link for the quiz and feedback form from yesterday, because I think a lot of participants could not attempt the quiz and could not fill the feedback form from yesterday. So I'll be sending that link again. Since it is compulsory for you to attempt all the quizzes, be sure that you complete the quiz for yesterday also. And in some time, I will also be sharing the quiz and feedback form links for today. And third is that tomorrow is, before the sessions that we have for tomorrow, we have a small networking and valedictory session for which there will be a link for MS Teams. I will be sharing that too. So look out for these 3 links that I will be sharing soon. So Shraddha ma'am, are you ready? I could just start with the introduction.
Yeah, hi.
Hi. So our next session is on non-omics data on Poly by Ms. Shraddha Dumawat, Elucidator, New Delhi. Shraddha Dumawat is a part of the technical pre-sales team functioning as a data analyst to provide biomedical ML solutions to prospects. She has been motivated to bring change in the healthcare industry and has always been interested in applications of machine learning and data science in medicine and drug discovery sciences. So I now request Shraddha Dumawat to please start the session.
Hi. Before I start sharing my screen, I would just like to have a scan of the audience. How many of you have experience in working with clinical trial data or non-omics data as such?
So if the participants do have experience, just type a yes in the chat box.
You're not audible.
I'm saying if the participants do have some experience, they can type yes in the chat box.
Well, okay. Anyways, I would share my screen now, and let's look at it then. So is my screen visible?
Yes, it's visible.
Um,
You can go ahead.
can anyone confirm if my screen is visible?
Yes, it is visible. You can go ahead.
Cool. So, so non-omics data basically includes various data types such as flow cytometry data or mutation data or data like lab measurements, just chemistry tests or immunology tests, blood cell counts, etc. That are usually a part of clinical trials or biological studies that happen in the process of drug discovery. So as you would know, life sciences R&D critically relies on 2 trillion GB of data generated almost every year. And while machine learning has a potential to unlock tremendous value for researchers through this particular data is very unstructured. So whatever you find on public sources such as patient studies or government agencies, from electronic health records, from proteomics data, is highly unstructured and non-harmonized, and it is very difficult to curate them and use them for analysis. So Poly's data infrastructure, as mentioned by the speakers that came before me, Anaga and Harsh, basically engineers and harmonizes the data at scale in order to provide you through three different portals, if I may say, such as programmatic access, GUI, or integrations. And this provides you with a standardized data schema. Anaga might have gone through both GCT as well as H580 schema that we use for various data types and single cell respectively. So our roadmap is basically cleaning biomedical data across all stages of drug discovery, that is from preclinical to post-approval research. So we go from preclinical research that is required in a drug discovery process to clinical operations, curating data at each and every step so that it's easy for researchers or bioinformaticians or biologists to go through this particular data and get insights and analyze data from the same. So today we are going to look at one particular data source called import. So the immunology database and analysis portal is one that provides advanced information for technology support. And this is this is a huge data warehouse containing experimental data and metadata describing the purpose of study and the methods of data generation. There are various types of data types that are available on import, from flow cytometry to PCR to ELISA to RNA sequencing to blood tests, etc. So there are various studies, and these are classified into various essays as such. So as you can see, this is basically the study model that is followed on import, a summary of the study model that is followed on import. And as you can see, there are various arms or cohorts that are made upon a clinical study or a design, and that subject is further categorized into a particular biosample, which are further given a particular treatment, or they go through a particular reagent, or have a HLA typing result, or various other assays that could be performed there. There are various lab test panels and lab tests that happen, which again go through the same route of going towards a biosample annotation and then having various studies or analysis performed or assays performed for them. So now I would like to go ahead and showcase how on PolyPython we look into a particular study of our interest. Before that, I would like to walk through the graphical user interface on Poly. So if you are logged in into your accounts, you can see that on import omics atlas, just can anyone confirm if they are logged in right now and if they're following the steps? I request you to please log in on Poly using the credentials that were provided uh, by Anagha and Harsh. Thanks. Thank you. So as you can see, this is the import omics atlas, and We have various data types such as lab measurement, proteomics, titer, PCR, cytometry, and we have various diseases that are covered in these particular studies. Right now, Poly hosts around 200,000 data sets, uh, all containing import experiment design studies or clinical trial studies. And if you can see, we have these particular samples uh, that are categorized as individual data sets. These are all very structured and harmonized, so it is very easy for us to run analyses and query the data on Poly. So I would like to go ahead and launch a notebook like through the workspace. So again, following the same steps as Anaga and Harsh did, we would like to go to the shared workspace, that is clinical trial, that is Amity Uni Workshop, and go to the notebook named clinical trial and copy it to your workspace. So yeah, kindly follow the step and launch the notebook with 16 GB. So I'm launching the notebook. Can anyone confirm if they have launched the notebook successfully? Okay, so anyways, so today we are going to, okay, it's still loading, fine, I'll wait. Just to give a background while we are waiting. So today we are going to look at a particular trial for the drug called lovastatin. Does anyone here know what lovastatin is used for? The drug? Okay, so lovastatin is basically a part of drug family called as statins, and these are HMG-CoA reductase drugs that are primarily used in diseases such as rheumatoid arthritis. Do we have the notebook loaded? Yeah, it is similar to, yes, thank you. Yes, exactly. It is a HMG-CoA reductase inhibitor, and it is used in various diseases such as rheumatoid. Cool. So it is used in various diseases such as rheumatoid arthritis, etc. So my teammates might have shown that they had downloaded various dependencies for the notebook. In the same way, we are going to install PolyPython. Please run the cells as I'm talking. So we are going to install PolyPython and then go ahead and install the libraries that are required for running our analysis. Clinical trial. I'm pretty sure everyone knows how clinical trials are actually structured. We saw a lot of them in the news during the COVID-19 vaccine rollout, right? So clinical trials basically go from discovery and preclinical stage to phase 1, phase 2, phase 3, and finally FDA approval. And patient participants basically receive specific interventions according to the research plan. And these interventions might range from medical products such as drugs and devices, procedures, and they basically look into the changes of participants' behavior or diet or various other immunological values that are more intrinsic in nature. So clinical trials may compare a new medical approach to an approach that is either already available or to a placebo, or a placebo is basically something that has no active ingredients as such, or to no intervention as such, and they would like to just look at how this particular drug has affected the patient cohort that took the drug as compared to the ones that did not or took placebo, basically. So I would like to go ahead and explain how the idea behind lovastatin. So this particular pathway describes the pathology of rheumatoid arthritis and how the statins counter it by inhibition of HMG-CoA reductase. So basically, statins by inhibition of HMG-CoA, reduce the level of inflammation, cytokine production, and T signaling, and stress and fiber formation, which leads to the lowering of symptoms in rheumatoid arthritis. So this particular study, that is SGY 473, basically looks into this particular mechanism and wants to understand the CRP values, that is C-reactive protein. So does anyone know what would, I mean, it's written out there, but what would CRP levels indicate? What would higher amount of CRP levels indicate? Well, so a higher amount of CRP or C-reactive protein, yes, exactly. So it basically indicates a higher inflation, and upon the inhibition of HMG-CoA reductase, one should see a drop in the inflammation level of people with RA. Hence, the logic of using lovastatin for rheumatoid arthritis. So basically, this is a double-blind, placebo-controlled randomized trial determining whether treatment with statin reduces the serum CRP in subjects with mildly active rheumatoid arthritis. So there were various time points or date points at which patients were screened to determine whether they were in the inclusion or exclusion criteria. So any clinical trial has multiple inclusion criteria. For example, the age should be of certain limit, and the person should not have any underlying diseases, or the weight should be of a certain from a certain bracket, etc. And they were given 80 milligram per day of lovastatin versus placebo of the same amount for almost 12 weeks. And so this is a very interesting study to look at, and hence we would use our clean, harmonized, and curated data to analyze the CRP levels of the clinical trial participants at different time points to understand the effect of lovastatin. So let us query data on poly. So if you were on import, you would have to on the source, you might have to download all the data and then use multiple functions to, combine the data and then use it further. On poly, we simply query for the data sets which are data set IDs, which are, correlated with SGY 473 study, so that we get all the samples that are, related to it. So that fetched around 1,547 data sets. That is 1,547 participants of the clinical trial. So the study basically looked into various different types of assays. So it looked into blood cell count, comprehensive metabolic panel, fasting lipid profile, hormone measurement, immunology test, protein or enzyme type measurement, and ELISA. So by looking into these different types of assays, they were trying to determine the effect of lovastatin in patients that took lovastatin versus participants that took placebo. And so since we are only interested in looking at the CRP levels right now, let us look at the immunology test as such. So again, I try to query data sets that are only correlated with immunology tests, and I find 192 patient data sets that are correlated to immunology tests. So I hope so the people who are on the notebook, can you just confirm if you have run these cells? I can wait. So poly, until we are waiting, poly basically allows you to create cohorts for a particular data set with one patient or one patient per assay per data set into one place, so that you could generate various analysis through it. Since the data is very clean and it is of a standardized type, it is very easy to merge the data and try to understand the various levels of the particular assay that you're measuring. So if you're measuring CRP levels here, it is very easy since all the patients are at one place and you can easily make a cohort of it. So, okay. So basically, can I move ahead?
Yes, I think you can move ahead. I think if the participants have doubts, they can use the chat box.
Are there any doubts?
If they have doubts, I think they can use the chat box, but I think you can move ahead.
Okay. All right. So I would request you to after clicking on the 3 cells above, I would request you to create a cohort by running this particular cell. And after the cohort is created, you can merge the data set level. So Harsh and Ananga might have explained how data on poly is curated at 3 levels. That's data set, sample, and feature. So you can further merge the data at data set level and sample and feature level. So as you can see, at data set level, we have information. We have information such as the biosample accession number that was showcased in the study model of import. And we also have the various research focus area, which is autoimmune. We have the cohort description, and we have other data set level information that could be useful. Apart from that, we also have sample level information. So sample level information basically looks into columns which describe uh, the planned visit accession, that is the date of visit. It is coded in this manner. And then the study accession, then study time to event, etc. It also looks into criteria such as ethnicity, gender, race, and age. So basically, this allows one to understand how there could be differential expression, or there could be differential levels of such particular proteins or any other assay that you perform in different ethnicities or gender in order to gain analysis through that perspective. So yeah, so arm name basically describes if the particular data set had a patient who was given lovastatin or who was given placebo in nature, or the drug or placebo in nature. And so yeah, then we have the feature level data set, which basically describes the result value, which is there. So here we can get the C-reactive protein result value, and we can understand by making various graphs, which I will show further, um, how it has affected through the period of time. So I see a question. How do I control for population heterogeneity if the results are coming from different geographic regions? So again, so you could you could modify the data in any way you want on the notebook, but you could also, so these are already planned out clinical trials or clinical studies that are there from one particular institute, and these are not randomized studies, right? So uh, so since the data is curated, what you could do is, if you're interested only in seeing one particular geographic location, you could curate it out, could remove that particular sample, generate different cohorts of it, and then analyze it. So that's the way you could deal with heterogeneity. So anyways, so then we could generate a box plot to understand the CRP levels over time. And so we have so the study was taken in such a manner that before the baseline day, 7 days ago, people were checked for their CRP levels, and then they were checked at the baseline level, and finally at the 84th day, where the treatment ended. And here we see that um, the placebo part of it do not see a rather major difference between the different days, that is -7 before the baseline and 84th day, while on the other hand the people who took lovastatin showcase a major difference between the amount of CRP levels between the baseline as well as 7 days before the baseline day and the 84th day, where we see a huge increase of the amount of CRP present in the participant's body. So this is how you could, generate insights from non-omics data using import through Poly. And if you have any questions, please let me know. It was great demonstrating this for you. Any questions?
I think there is one question. So Deepti, can you take it?
Yes, ma'am.
Last slide, as in the last image, is it? Can you please confirm if it's the last image?
The last slide.
Is it, are you talking about this one? Okay, yeah.
Yes.
Cool. So, just a minute. Yeah, okay. So basically what happened in this particular clinical study is that there were multiple different time points, but now we are going to look at 3 major time points wherein they took into consideration of maximum participants. So what they did was before the clinical trial started, or before the administration of the drug started, 7 days ago, they took the assay of CRP levels. I mean, they measured the amount of CRP levels in a particular patient or a participant. And then on the baseline day, they again, measured the amount of CRP levels that are there in the participant. And then they started their treatment. And on the final day, they wanted to check how much CRP levels are there if they kept giving 80 mg of lovastatin every day, or based on every visit of the patient. And not every day, based on every visit of the patient. And finally, on the 84th day, they wanted to understand what would be the difference between the -7th day, the baseline day, and the 84th day, that is the final day of the clinical study. And what we can understand, the insight that we can generate from this box plot is that there was a significant decrease in the amount of CRP level. Even after running the code to generate cohort. Did you, were there any errors?
So Burokha, if you face some error, you can type that.
These are not 2 groups. This is the median. Are you talking about this? Just to clarify, Burokha, if I'm pronouncing that right, were there any errors in the notebook that you ran? No, okay. So you could connect with me later probably to understand this. Jitendra, to answer your question, do you mean in the blue box itself? Then this is not 2 groups. This is the median. This is how a box plot is measured. If I'm understanding your question right. Yeah, of course, other plotting libraries could also be used instead of Plotly. And yeah, so you can find the raw data on import, and you can use your own methods of data cleaning, if you will, or you could download the data from Poly as per your subscription, and you can use it. But they are from Poly, you would only get it in GCT format, which is a much structured and curated format. And you could use the GCT format by parsing it, and you could generate the same insights from it.
So any other doubts?
Great.
Or
Any other questions?
so I don't think there are any other questions. Thank you so much for joining us. It was a really good session and we really enjoyed the way you shared all the details, and I'm sure the participants learned a lot.
Good.
I will
So,
also take this opportunity to thank Team Elucidata, all the team members who joined us throughout the day. It was an excellent session and we really enjoyed the hands-on session also. It was very descriptive and I hope the participants must have understood a lot about the data cleaning and all the procedures that were discussed.
great. Yeah, sure. So this is my mail ID and you could reach out to me to clarify your doubts. Great, thanks a lot.
Thank you so much. So Sudipti, I think we can go ahead. Bhavna ma'am is already logged in.
Yes, ma'am. So our next session is on omics data integration tools and databases by Dr. Bhavna Rakhi ma'am, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida. Dr. Bhavna Rakhi ma'am pursued her PhD at Gautam Budh Technical University and worked on an in silico study of hepatitis E virus using bioinformatics tools and techniques. She is the co-guide in DBT's twinning program for the NE titled Association and Polymorphism of HLA class 2 alleles in rheumatic heart disease patients of Northeast India. And she has been the recipient of Department of Science and Technology Young Scientist Award to attend an international conference on vaccines in the US. I now request Dr. Bhavna Rathi ma'am to please start the session.
Thank you, Sudipti. Good evening, everyone. Let me share my screen. I hope my screen is visible.
Yes, it is.
Okay. So basically the topic which Sudipti referred is, this is a different topic on which I have prepared the presentation. And this is reverse vaccinology. This is one of the most exciting areas because we have been under the pandemic for almost 2 years. So I just thought this is a more relevant topic. So this is a reverse vaccinology, and basically what reverse vaccinology is, it's an advancement in vaccinology which generally employs all your bioinformatics techniques as well as the reverse pharmacology practices. This particular technique was being pioneered by Rainer Rupiolo, and this particular technique was first being used on serotype virus B, that is, serogroup B meningococcal vaccine. Now, the main objective of this particular study, or the reverse vaccinology approach, is the other name which we even use for this particular vaccinology approach is in silico peptide-based vaccine designing. This basically minimizes the laboratory-based research. So the wet lab experimentation time would definitely be minimized if we are using this computational method. We can easily develop, do the computational analysis on the antigen using these bioinformatics tools. I would be demonstrating a few of tools with reference to this particular approach. And eventually this is going to help us for the development of immunodiagnostic tests and vaccines. This particular approach is being widely used for on various diseases, infections, allergies, autoimmune diseases, as well as on various cancer types. So first of all, we need to understand what is the role of reverse vaccinology. So, and what exactly does the term stands for. So the basic idea behind this approach or reverse vaccinology is that we would have the entire pathogenic genome, which is going to be screened using various bioinformatics approaches to find the most suitable gene, which can be served as a good vaccine candidate. The gene which we are looking forward for, the protein which this particular gene should code should be preferably an extracellular localized protein. And eventually we have to map this entire protein to identify the region which is going to elicit a good amount of humoral immune response as well as cell-mediated immune response. As I just mentioned, in early 2000, this particular scientist has pioneered this approach for the very first time. And the attempt of this particular reverse vaccinology first began with the meningococcus B, that is meningococcus serogroup B. So nearly over 50% of the meningococcal meningitis was because of this particular serotype. And none of the efforts were being successful for the by the scientists in this particular approach. And the reason was that the bacterium polysaccharide shell was highly identical to the human self-antigen. Now, to overcome this particular approach, what the scientist group did, they tried to identify and they start exploring the various surface proteins. And considering these surface proteins, they have developed this and they have used the bioinformatics techniques to design a functional vaccine. This next slide is a very good depiction where you can easily see the two pipelines wherein one is the conventional vaccinology approach versus the reverse vaccinology pipeline. As we can see, the first pipeline, if we are working on a conventional vaccinology approach, this is the microorganism we have to cultivate or we have to culture that. Eventually, we have to select the antigen. We go for the immunogenicity component purification. We go for cloning of genes, testing in the animal models, and then we go for the phase of development of vaccine, ultimately leading to a vaccine. So this whole process is going to take a relatively more amount of time as compared to the reverse vaccinology approach, where we directly work on the genome sequences or the proteomic pool of the pathogen. In considering this proteomic pool, we have to look ahead for the prediction of the novel antigens. We have to check that that particular antigen should be non-homologous to the Homo sapiens genome. Then we are going ahead with the recombinant protein expression of that particular protein. Eventually, immunogenicity testing in animal models, the development phase of the vaccine, and the vaccine comes into consideration. So this is the working pipeline for the reverse vaccinology approach. Here in the next slide, I have just mentioned, this is a pictorial representation which has been taken from the first paper which came in scientific reports in early 2020. That was 2020 when the pandemic started. So this is a approach which is used for a candidate multi-epitope vaccine against SARS-CoV-2. So what they did in this particular same approach has been used here for SARS-CoV-2. So the entire proteomic pool of SARS-CoV-2 was being considered. And after the consideration, the second step was the selection of the virulent protein. Now, the virulent protein has to be identified from the entire proteomic pool. And whenever we are working for a peptide-based vaccine or epitope-based vaccine, we generally prefer to have a surface antigen or maybe an outer cellular membrane protein. So in this case, this spike glycoprotein FASTA sequences were being retrieved from PDB. And this is an additional study which they performed, that they have even done the phylogenetic analysis of the selected proteins, but somehow it has no role to play with the reverse vaccinology approach. The next step was to identify all the cytotoxic T lymphocytes, helper T lymphocytes, and IFN gamma epitope prediction. So this is a very important component of the reverse vaccinology approach, wherein we are using various bioinformatics methods, algorithms, to identify and map those particular regions of the antigen which has the potential capability to have an effector cell-mediated immune response. The second thing was prediction of these binding of these CTLs and HTL epitopes with MHC class 1 and MHC class 2 alleles. So this is a very important component here. Even if we identify the cytotoxic T lymphocyte and helper T lymphocyte epitopes, they would definitely not have a very good effector potential capability until and unless they're very strong binders with MHC class 1 and class 2. Because eventually these would be the cells which to which your T cell receptor molecules would identify. So once these epitopes have been identified as strong binders with MHC class 1 and class 2 alleles, the multi-epitope vaccine construct was being developed. And once this multi-epitope vaccine construct is being developed, so this particular vaccine construct just has the potential capability of eliciting cell-mediated immune response. The second leg of this particular work is to identify the humoral immunity, and we have to identify the B cell prediction has to be done on the vaccine construct, wherein we have to identify both the discontinuous B cells as well as the continuous B cells. So once we identify these continuous as well as the discontinuous B cells on this vaccine construct, then we are very sure with the fact that now these epitopes which have been which has been identified from the whole of the epitopic pool which has been generated by all these mapping techniques, we can finally zero down on some important 7 to 8 candidates which can actually work as a good vaccine candidate. So once we have this vaccine construct, the next step is going to be the allergenicity and antigenicity prediction of the vaccine, because we need to check whether this particular vaccine construct, as this is not a same format of the spike glycoprotein anymore. So we need to check whether this new combination or this new concatenation of this chimeric protein is going to be non-allergic as well as non-antigenic at the same time. And eventually it should be non-toxic as well. So the toxicity test is also being conducted. Next step is the physiochemical prediction of the vaccine. We have to check all the physiochemical parameters. Next step would be we have to go for the tertiary structure prediction, secondary as well as the tertiary structure prediction. And we can use various techniques to do the secondary as well as the tertiary structure identification. Within this step, the structure is ready and we have the final vaccine construct ready with us, which is based upon the reverse vaccinology approach. And now this particular vaccine, we have to check that what is the docking capability of this vaccine with the toll-like receptor. So this study has done this docking test with the toll-like receptor 2, toll-like receptor 4, and MHC class 1 and class 2 receptors. Because if we have a good binding affinity with these receptor molecules, then it gives us a very clear picture that these epitopes would definitely have a very good effector immune response. Once we have done the molecular docking of these epitopes with the receptor molecules, then we have to do the molecular dynamic simulation of this vaccine construct to check the potentiality and the stability of this particular complex. The next step is the reverse translation and codon optimization, which has been done through JCAT and SnapGene tools. Generally, it is to check the codon optimization bias as well as to do and finally we do the in silico cloning of this particular vaccine, and we go ahead with the immune simulation. So there is a tool by the name C Immune Sum, and this is a very nice tool which does this immune simulation, wherein you can decide the dosage of your vaccine, and then you can even tell after how many number of days you are going to administer this injection of the vaccine, and eventually how exactly the titer of the antibodies is going to be. So this is the entire reverse vaccinology approach, which depicts the case study in the case of SARS-CoV-2. Now, these are few very important and tools which are very readily being used. These are some computer-aided bioinformatics projects, as we can see, which are employing this technique of reverse vaccinology. One is the NERV tool. This is the NERV server. Then we have Vaxygen. It's a very comprehensive program, which is very much easily publicly available. It's in the public domain. You can easily and efficiently use this. The results are also very promising. And then we have this tool by the name Rank Prep. So this is an online program which helps you to identify the peptide bonding to your MHC molecules, and the process which it involves is based upon the PSSM matrices, position specific scoring matrices. Now, these are certain epitope prediction programs. So basically an antigen, if we say this is the substance which is going to be recognized by the antibody, the receptors of B cells and T cells. And in case of T cells, these particular epitopes have to be in a binary complex with the MHC molecule. So epitope prediction has to be done very accurately so that we get to know eventually that they have the potential to elicit a good effector immune response. This is just this slide depicts the role of epitope prediction and reverse vaccinology approach, which is just previously discussed in the previous slides. The major role of the immune system is, as we know, we have to have a good trigger of the B cells and as well as the T lymphocytes. So the prediction of these epitopes plays a very important role in determination of the candidate vaccines. Various approach which we are using these days is they're based upon ML and AI techniques. We have artificial neural network programs and machine learning programs. Various ML programs, we are using this random forest as well as SPM methods as the baseline algorithms for predicting these epitopes. So this particular slide is being bifurcated into 2 steps. So this is one of the methods, major methods which we use for B cell epitope prediction. One is the structural method. So where we have the structure of your antigen and antibody, which we look through X-ray crystallography, NMR techniques, electron microscopy, and based upon these antibody antigen complexes, based upon this experimental data, we are making predictive algorithms for B cell epitope prediction. The second method for doing B cell epitope prediction is through the functional approach, where we are using various mass spectrometry as well as immunoassays like ELISA, ELISA plot, and Western blot. Now, this slides give us the information for two major servers which are being highly used for discontinuous B cell epitopes. So one is the Discotope tool, which has been very prevalently used for identifying the discontinuous B cell epitopes. The other one is the ELIPRO program, which is also one of the major tools which has been which has a very good efficiency, which has been used for predicting your discontinuous B cell epitopes. And ELIPRO is having an upper edge because it has the potential to even elucidate the linear. We can even identify the linear or continuous B cell epitopes using the ELIPRO server. And these are certain continuous epitope prediction programs for B cells, but generally it has been always seen experimentally, it has been verified that discontinuous B cell epitopes has major potential of eliciting the humoral immune response as compared to the continuous B cell epitopes. But these are few methods which are very prevalently used based upon the antigenic protein amino acid property, like the Chow-Fasman beta turn method is there. We have the surface accessibility prediction. There is a flexibility prediction method, and there is an antigenicity as well as the hydrophilicity prediction method. So basically, all these methods which are being employed for identifying the continuous B cell epitopes are based upon the antigenic protein or amino acid sequence. Next in line is various important T cell epitope prediction programs. So this particular slides just tell us that the diagnostic pattern which we use for T cell epitope prediction is generally immunoassays, like we do ELISA or we do the Western blots. But to avoid these wet lab approaches, because it generally takes a lot of time, so this particular technique is just to have the leads in hand, and then we can directly go for the chemical synthesis for diagnosis or maybe for a peptide-based vaccine designing. So these are few tools which are being very prevalently used for T cell epitope prediction. And this particular image I have taken from the IEDB browser, wherein we have these tools for MHC. We have this T cell epitope MHC binding prediction, peptide binding to MHC class 1 molecule, peptide binding to MHC class 2. Then there is a TEPI tool, which has the potential for checking the binding prediction with both MHC class 1 and class 2 tool. There is a T cell epitope processing predictions also. We have a tool which is proteasomal cleavage tap transport MHC class 1 combined predictor, which gives us very promising result as compared to the regular MHC class 1 binding tool. And then there are various tools like neural network prediction of the proteasomal cleavage, that is by the name NetChop, and T cell epitope, that is NetCTL and NetCTL packet approach. Now, these are certain, this particular slide is basically telling us the pros and cons of this particular approach, that is reverse vaccinology. This is a highly challenging task if we are doing it theoretically. This particular approach is in silico. The process is really fast and efficient. The complete your vaccine designing can be done in a very short span of time as compared to going ahead with the wet lab experimentations. So eventually we can identify those epitopic sequences which has a high efficacy of creating or giving a good effector immune response, combining your cell mediated as well as the humoral immune response. And eventually we can take up those few epitopic sequences. They could be synthetically, they can be chemically synthesized, and eventually the study can be taken to wet lab. And for the detection of these antibodies, we are using this particular technique for infections, allergies, autoimmune diseases, as well as various bacterial diseases, viral diseases, and even cancer. But the only disadvantage of using this particular technique is that we can only work on protein sequences. So this is one of the major lacuna we have whenever we are discussing this particular approach, because even if we are looking for some non-peptidic biomolecules, then we cannot target the reverse vaccinology approach. And this slides give us the recent advancement. The various research efforts are actually underway to develop the synthetic peptide-based vaccine on HIV, on human T cell leukemia, virus type 1, streptococcus pyogenes infection, malaria, SARS, and synthetic peptides are also being into consideration for a therapeutic vaccine for cancer, Alzheimer, and autoimmune diseases. So this was just the gist of the entire description, what exactly reverse vaccinology is, what is the working pipeline for reverse vaccinology. I'll go for the hands-on demonstration. So this is the IEDB browser. So this is the IEDB browser, which is a very important portal for immunological information. The browser is immunoepitope database and analysis resource. As you can see on this particular page of the browser, it has two major sections. In two sections, one is the interface for the database, where you can identify or you can look into various epitopic sequences which have been performed through experimental analysis. So if we want to look into some T cell assays, B cell, MHC ligand, we can identify. And the other section of this browser basically involves various epitope analysis resource. As we can see in the summary matrix of this browser, the epitopic peptides, these peptidic epitopes are around 15,37,000. So it has a huge range of these peptidic epitopes which are prevalent. But there are around 3,000 something epitopes which are even non-peptidic or even for biomolecules. It has done a total of around 4,43,000 T cell assays. The B cell assays around 13 lakh. MHC ligand assays is around 46 lakhs. And the complete source organism for which the data is existing on this particular portal is around 4,226. And the references, as we can see, there are around 23,000 references. So this was just a summary matrix to give a glimpse of this particular browser. Now, suppose I have to identify information regarding any of the particular infection. Let's go ahead with SARS only, because this first section is if I have to look for epitope, I can conveniently look into the linear epitopes or the discontinuous epitopes if I'm considering in the case of protein, and even I can go for non-peptidic. If I am not looking for a protein epitope, I'm looking for some other biomolecule, maybe a polysaccharide or something. But I'm keeping it at the default setting because I need to know as many number of epitopes which have been experimentally validated in the case of SARS. The next section here, as you can see, this is the organism name. So herein I can type SARS. So this is SARS-CoV-2, and this is the organism for which and the ID is being referred. So I can easily identify through this particular organism. This is the epitopic source. Now, if I'm specifying the antigen, maybe I know that I'm just looking for antigen which is based upon the spike glycoprotein. I can write the antigen name here. Maybe I'm targeting the core protein, I'm targeting the capsid or membrane protein. But in case I'm keeping this particular section blank, so it is going to give me the entire epitopes which are existing in the case of SARS-CoV-2. And the host is obviously human. We are looking for a human infection of COVID-19. And I am initially just looking for B cells. So let's see how many B cell epitopes are existing here for COVID-19, and the disease section I can switch to infection, allergic, depending upon. So I'm keeping this as the default setting. So the current filters which we have used for this specific search is the organism is SARS-CoV-2, that is COVID-19, which should include all the positive assays. We are not considering the T cell assays. No MHC assays are there, and the host which is going to be targeted is human. So here we can see that we have achieved around 8,905 epitopes, specifically B cell epitopes, which triggers the humoral immune response for the SARS-CoV-2, and this is the epitopic IDs there. Total of 16 antigens have been identified here. So if you can see here, we have epitopes for the spike glycoprotein, we have epitopes on the nucleoprotein, membrane protein, and the list of all the proteins of SARS have been mentioned here, against which we have these antigens. So a total of 16 antigens is being given. If I need to specify a specific protein here, I can specify that as well. This is the total number of assays, which gives us the description of the epitope with the ID, its reference, the sequence of the epitope, that particular antigen on which the epitope is present, the assay description is mentioned just against that. And it also refers whether it was a positive assay or a negative assay. This section is the receptor. So it gives us the information against all the receptor region, which has the binding affinity of this B cell receptor. And these are the references which we can see. Various journals are being mentioned with their PubMed IDs. So let's open one of these IEDB IDs. So this is the epitopic summary. As we can see, this is E484, that is the number of the amino acid. This is a discontinuous epitope. This is the ID. And this has been studied on the spike glycoprotein for SARS-CoV-2. The epitope has been studied for immune reactivity in 11 publications, and this particular epitope has been tested on 95 B cell assays. So similarly, you can just play with these parameters to look for more details. Suppose I am just interested in linear epitopic sequence, so I can select linear here, and I can just modulate the search. So this time I will be getting only those particular epitopes which are linear in nature, and I will not get those epitopes which are discontinuous. So we can see the change in the numbers. Earlier it was 8,900 something. So right now it is 8,204. Similarly, we can even look for the non-peptidic ones, and we can even change and see if I want to look for T cells, I can select T cell here. If I'm looking for linear, and I can even specify the length also. Maybe I'm targeting a particular length. So I can specify the length. Maybe I'm looking for 8 mer, 10 mer, 11 mer, or 15 mer epitope. So that can be conveniently done with these interfaces. So this was just a glimpse of the database, how exactly the database searching has to be done on this particular browser. Next information which we have on this particular portal is regarding the epitope analysis resource. This is the first segment of the tools, which is T cell epitope prediction tools, like we have for MHC class 1 binding. Now, this is the interface for MHC class 1 binding prediction. So if I have to predict any, I need to just have my protein sequence, my antigenic protein sequence, or the virulent protein, which I have selected from a particular virus, bacteria, or maybe a tumor type. I can simply paste that sequence here, or maybe I can upload from this particular section. So I'll do one thing. I'll take the example data set from here. This is the data which they have given for SARS spike protein. So automatically it is going to incorporate the sequence of SARS spike protein here. Next thing, what I have to do is I have to check for the prediction method. What exactly is the method they are using? So this is the net MHC pan method. And there are other methods also, like ANN consensus method is there, which is a consensus of various methods. So I can just change these methods depending upon my convenience, and checking the accuracy, which method is, or which algorithm is much more accurate. The MHC source species obviously human, because we are targeting the human infection. So we want the association of this spike protein with the human HLA. And eventually we can even change these HLA types as well. But right now I'm just concentrating on the human as the host. And then this is the allelic. I have to specify on which particular allele, for which particular allele I'm doing the prediction. So suppose I just pick up HLA A0201. And eventually I have to specify the length also. So if I'm targeting MHC class 1, obviously the length is going to be somewhere between 8 mer to a 10 mer, because the MHC class 1 group supports a binding capability of 8 mer to a 10 mer. So I'm specifying here as a nanomeric sequence. And I can add as many number of alleles which are required and specify the length accordingly. Now it has a very important thing, which is, if you can see, the beauty of this particular program is that you, even if this allele is not existing within their database, you have an option to upload your allelic files. So many times if we have a population data set with us, maybe we have a northeastern population data set, maybe we have some population data set of some specific geographical location that not might be a part of their database, but still that allelic file can be uploaded. And conveniently, we can check whether the epitope or that particular virulent protein is going to have a good binding affinity with that particular allelic frequency. The output is going to come in this format. We can sort the peptides either by their scores, or we can even sort it on the basis of their positions in the sequence. So I'm just keeping it as on the default setting, and this is completely optional, your email ID, and then you just have to submit. Now this is the result. As we can see here, the first thing is the sequence which we took, that was the spike protein of SARS. And here, as we see, I have just considered two alleles here, that was 0201 subtype and 2301 subtype. The closest alleles, because this was the input which I have given, and the closest allele which is here is HLA 0201. So the distance here is 0.00. So why the distance is 0.00 is because I have just picked up the alleles which are already a part of this particular database. I have not given my allelic data set, otherwise it would have looked for the closest allele. So obviously there is no distance, that is completely zero. And next is the file. That's the table. So this is the result. We can download this in Excel and we can do the further the data can be processed, the allelic frequency, the start of the epitopic sequence, the end of the epitopic sequence, the length, the peptide, the core region score, as well as the percentile rank. So this is for MHC class 1 binding. Similarly, we have a tool for MHC class 2 binding. This is almost the same process. This is exactly the same. I'm not going to run, I won't run this particular tool, but this is exactly the same. You can take the example sequence, just paste your sequence here. You can change this program, and there is a tutorial, there's a help file which has given a complete description of all these methods which have been used and what is the accuracy rate of these methods. Eventually, we can specify the allele here. So as this is class 2, so this is HLA, DR, DP, DQ, we can check among those. And then we can even upload the allelic files. And as it is MHC class 2 binding, so obviously the region which is going to bind because of the open groove structure is somewhere between 12 to 18. And we can just run and we would get the result likewise. Now, these are two important tools for T-cell epitope prediction or binding of the epitopes or these epitopes to your MHC class 1 and class 2 alleles. The next one is the B-cell epitope prediction program. So this is the first section is the prediction of B-cell epitopes using the antigenic sequence properties. And the next one is the prediction using the discontinuous B cells using the antigenic structure. So if we are using the first one, if we are predicting a continuous B-cell epitope, then we would be requiring a protein sequence. We can directly enter the Swiss prot ID here. So I am just considering this examples, which has been already mentioned here. This is the Swiss prot ID. We can enter the accession number or we can paste the sequence here. And then these are the various methods which we can use for predicting the continuous B-cell epitope. So this particular epitope has the potential for eliciting the humoral immune response. So if we see here, there are several methods like the Bepi-Pred linear epitope prediction, and this is one of the finest method with the highest accuracy, and this supports this is based upon the machine learning approach. And rest of these methods are based, as I have mentioned in the presentation also, this is the Chow-Fasman method, flexibility method based upon the hydrophilicity or the antigenicity of the protein. So we have just considered this particular protein and then we can submit. Meanwhile, till we'll get the results, let's get back to the next one. And the next one is the prediction of the discontinuous B-cell epitopes using the antigenic structure. Now, the difference between two different versions of the program for discontinuous versus continuous is for the continuous B-cell epitopes, we can just play with the primary protein sequence. But if we are working on the discotope method or we are focusing to identify discontinuous B cells, then we would be requiring the antigenic structure. So the antigen structure can be easily downloaded through the PDB database. And if the PDB database is lacking any antigenic structure, then obviously we will have to go for the tertiary structure prediction, considering any of the methods. We have to just mention the PDB ID here, like as this ID can be, this is the PDB ID. And along with that, we have to enter the chain number here. So as I have not entered the chain number, let me put it here because I myself doesn't know this protein is having how many chains. Okay, meantime, we have got the result for the linear B-cell epitopes by the Bepi-Pred method. This was the protein sequence which has been considered. And this is the kind of a graph you will get. So this is the threshold. And above this threshold, all these yellow peaks, as you can see, these are all your epitopic sequences, the linear B-cell epitopes. And the green peaks, which you are seeing here below the threshold, are all the negative results, which are non-epitopic sequences. Along with the graph, you will get a table or a tabular result as well. And this tabular result tells us that the first one is starting from 5 to 10. So this is the epitopic sequence where the length is 6. 38 to 61, this is 24 amino acid. This is a linear epitope, and likewise it has mentioned 6 B-cell epitopes on this entire length of the antigenic protein. This is how you will get the result for the linear B-cell epitopes. And this is the result for the discotope method. So this is all these green peaks are giving us the result for these are the positive results, which are epitopes for discontinuous B cells, and all these pink peaks are the negative predictions. and we can have a 3D view as well. So if we click on this 3D view, we can have the 3D view of this particular result. It is going to show us the antigenic structure, and along with the antigenic structure, I think this application is blocked here. But if you click on this 3D structure view, you will see the antigenic structure, and with the space filled model, you will see those epitopic sequences where the epitope is binding with that particular region would be reflecting. This is the region. This is the entire antigenic sequence. It is having 2 chains, and we have only done the prediction on chain A. So this is the blue portion is the chain A, and this is the region which is a discontinuous epitope. All these yellow region is depicting the epitopic region of the antigen. If we keep a cursor on any of these points, as we can see, it highlights the amino acid, and it tells us the position as well. The other tool which we have is the ELIPRO tool. This is almost the same like the discotope method. So if we have to predict the discontinuous B cell epitopes, again, we would be requiring an antigenic structure, and we can do it with the help of the ELIPRO program. Now, this is one of the most important section, that is epitope analysis resource, in which population coverage is a very important tool. So basically, what is a population coverage? Once we have done the prediction for these MHC class 1 and class 2 binders, we have identified these epitopes. Now we need to check that in which particular population they would have a good effector immune response. What is going to be the immune response in various population data sets? So basically this tool, what it has done, they have divided the entire world population into 16 geographical locations. And based upon that, we can check that, okay, if this is the epitope and these are the binding MHC alleles, what is going to be the efficacy, that how much effective this particular epitope is going to be in a specific population. So you can change the number of epitopes here depending upon how many epitopes you're checking. Maybe you have just identified top 6 leads, so you can write here 6. Right now it is just by default, it is 10 here. And I'll take the example data set from here itself. So I'm taking these 34 MHC class 1 restricted epitopes, because this is the epitope which we have identified from the previous programs for MHC class 1 binding. We would select MHC class 1 separate. We can even do a combined prediction. We can do separate prediction for class 1 and class 2. And by default, I'm just clicking the world population just to check that what is going to be the efficacy of these epitopes. If I take these epitopes, how much efficient they are going to be for showing a good effector immune response. So if you can see here, this is the epitope sequence, and these are the binding MHC alleles. These are all MHC class 1 alleles because I have selected that MHC class 1 option. And this particular programs becomes very important, maybe because if we are targeting a particular vaccine, the vaccine is quite effective in the Indian population. Maybe it is not that effective in your European population or African population. So to have a universal vaccine, this is the best approach, that we can check whether those alleles are being, those alleles which are existing in the other populations also are being targeted. So we would just submit here. This is the submission. So as we can see here, we have just selected this for class 1, and the coverage is coming around 95.6%. So on an average, this is the coverage that nearly 95, if this particular vaccine, these epitopes are being considered for making this vaccine under this reverse vaccinology pipeline. So they have a potential of eliciting a 95.6% of immune response. Likewise, you can do this change. We can go to the example set again, and this time we can take the data set, which is combining both MHC class 1 and class 2. So eventually, when we are looking for a vaccine description, we prefer to have both the set of alleles for class 1 binders as well as the class 2 binders. So these are the sequences. And once we can even select a specific population like Northeast Asia, and we can check there. I have to select this MHC class 1 and class 2 binders. Now this tells us that when we have taken this combination of 6 epitopes for class 1 as well as the class 2 binders, it is showing us a coverage of 96.11%. So this is a very good browser for various immunological resource. Either you are doing some kind of analysis, or maybe you are identifying just experimental data which is pre-existing on the database. And it has various other tools also, like the conservation across the antigens, the cluster within the similar sequences. So you can even take the various isolates or strains of a particular virus or a bacteria, and then you can do the consensus mapping, and then even you can go ahead with the cluster mapping, and you can identify the epitopes. Now, these are certain tools. There are three important tools as a working pipeline. This is one is the Vaxygen server 2.0. So if we have a protein sequence in hand, so we can just check the antigenicity of the protein, because if the protein is not antigenic in nature, it is not going to have a immune response. So we have to just paste the sequence here in the text box, and then we have to select the target organism, whether it is a we are working on a bacterial vaccine or a virus, maybe a tumor, or it is some parasite or a fungal organism, and then we have to set a certain threshold value, and eventually it is going to give us the result for the whether it is going to give us the result in a way whether it is a protective antigen for further studies or it is a non-probable protective antigen. So if we get a result, it is a protective antigen, then we can select that virulent protein, we can take that antigen and we can go ahead with the prediction. And even that pipeline, as it's referred, there was a vaccine construct which we have made. So we have to check the antigenicity of this vaccine construct on this particular tool, that is Vaxygen. The other tool we have is the Allertop tool. So this particular tool, Allertop, is to check the allergenicity prediction. So once we have the construct with us, that's the combination of these epitopes, we can use that particular protein sequence here and we can check the allergenicity. So eventually it should not be allergenic in behavior. If it is allergenic, then it is going to cause an allergic response to the host, and that is not going to be a safe vaccine for the host. And the third important tool which we have to use for the vaccine construct is the ToxinPred. This is the ToxinPred program, which helps us to do the protein scanning. There are various options of batch submission, motif scanning, but this one is the most important one because you can paste your entire antigenic sequence here before the vaccine construct sequence here, and we can identify the region which is toxic in nature. So we have to be very sure before we go ahead with the wet lab experimentation or we go ahead with the synthesis of these epitopes that neither of these amino acids should be toxic in behavior. So these are three important tools that has to be run. One is the allergenicity, antigenicity, and the toxicity. So that's all for the demonstration. I can take up the questions. And this is a small hands-on session. So you can just practice this. You can retrieve the cell surface binding protein of the monkeypox virus. So this is the self-surface binding protein because we have this monkeypox infection, very prevalent these days. You can take this particular protein sequence through NCBI or UniProt, GenPept of NCBI or UniProt database. You have to check for the antigenicity, allergenicity, and toxicity of the protein, map the continuous and discontinuous B cell epitopes, and then eventually identify helper and cytotoxic T cell epitopes, and then eventually perform the population coverage analysis of these top lead epitopes to check that they have a good potential of having an effective immune response.
Participants are encouraged to ask their questions through the chat box.
Sure, please.
I think we can paste this in the chat box, ma'am.
These questions?
Yes, they can take it down and do it. So I don't think we have any questions for now. And Bhavna ma'am is at the Center of Computational Biology and Bioinformatics. You can reach out to her in case of any queries. We can share, ma'am, you can share your email ID as well. Yes, so if there are any questions later, ma'am can be reached out at the email ID also. So I think, thank you so much, ma'am, for taking out the time for this session. And in case anybody reaches out to us also, we'll get back to you.
Thanks.
Thank you so much, ma'am.
Thank you, everyone.
All right, to the participants, I will just give some instructions. First, there has been links shared in the chat box for the quiz and the feedback. Please attempt the quiz as well as the feedbacks, because as far as the report I am getting, everybody is not attempting the feedback and the quiz. So please do that. That is the first suggestion I would like to give. The second thing is that, as Sudipti has already declared, there is a session which is going to happen from 12:30 to 1 tomorrow, which is on Microsoft Teams, and the link for joining the Teams is shared in the chat box. So you can click the link and you can join. Okay, so Sudipti, you can re-share the link of the quiz once.
Yes, ma'am.
And so please save this link for joining the teams also tomorrow at 12:30. All participants from Amity University or AIB are requested to be present in 406 for the session at 12:30, J3 block, room number 406. And for the rest of the participants, we will have the session online. So it is a kind of networking session and also where you will get the summary of the workshop held so far. Also, for my session tomorrow, I have uploaded a file which is su_cells.zip. I would request the participants who would want to run along with me the analysis that we'll be doing tomorrow to please download this file as well as have on your system R 4.2.1 for Windows. That will be the easiest way to approach the language that we are going to be running tomorrow. So you can just download. It is a simple download process. You can download it on your system as well as you can download this file if you want to run along the R code with me. That will be easier for us to do it because the downloading of the file and downloading of R on your system will take some time. So that's all from my side. I think we are good to close.
I would ask the participants to please save the link for the Teams meeting. Because
as
this
well
chat
as
will
the
be
links
down
for
after.
quiz and feedback also because these are important links. So please share
And
all
they
your
will
quiz.
move on once the meeting is over.
Yeah, chat box will be vanished once this meeting is over. All right. If anybody wants us to wait for 5 minutes, please write it in the chat box or else we'll close the session. 5 minutes. Okay. So we'll give you 5 minutes. Yeah, we'll wait for 5 minutes and 5 to 10 minutes maybe, and then we'll close the session.